Imagine a predator so ruthless that it didn't just hunt other creatures, it hunted its own kind. Meet the cannibal dinosaur, Majungasaurus, a terrifying hunter that roamed the ancient landscapes with a hunger that couldn't be satisfied by ordinary prey. Fossil evidence reveals bite marks on bones of its own species, proving that survival sometimes meant turning on each other. What makes it so terrifying? Before we dive in, let us know where in the world you're watching from in the comments below. It's always amazing to see how far our videos reach. And if you enjoy learning about the prehistoric era, consider subscribing. It really helps us keep making more content like this. Majungasaurus was a remarkable predator that roamed Madagascar between 70 and 66 million years ago during the final chapter of the Cretaceous period. This places it among the very last non-avian dinosaurs, living right up until the catastrophic event that wiped out most dinosaurs at the end of the Cretaceous. Today, scientists recognize only a single species within this genus, known as Majungasaurus crenatissimus. At one point, this dinosaur was also called Majungasaurus, but that name is now considered a later synonym and is no longer used. As a member of the Abelosaurid family, Majungasaurus shared many characteristics with its relatives. It walked on 2 strong legs, had a short snout, and sported extremely tiny forelimbs, which were so short that they seem almost comically small compared to its massive body. Its hind legs, by contrast, were powerful and stocky, built to support its considerable bulk. Adult Majungasaurus could reach roughly 7 meters in length, about 23 feet, and weigh more than 1 ton, or 1.1 short tons. One of its most striking features was its wide skull, covered with thick, roughened bone on the top of its snout. This included a single rounded horn on the crown of its skull, a feature so unusual that it was originally mistaken for the dome of a pachycephalosaur. Another notable trait was its teeth. It had more teeth in both the upper and lower jaws than most of its Abelosaurid cousins. Majungasaurus is also historically significant as one of the first Abelosaurs to be discovered. Its first remains were unearthed in the year 1896, although at that time they were thought to belong to the genus Megalosaurus. Over the decades, the fossils were repeatedly re-examined, and it was not until 1955 that the genus Majungasaurus was officially named. Thanks to several well-preserved skulls and abundant skeletal material, this dinosaur has become one of the best-studied theropods from the southern hemisphere. Interestingly, studies suggest that it was more closely related to Abelosaurids from India than to those from South America or mainland Africa, a discovery with intriguing implications for how these predators spread across ancient continents. In its ecosystem, Majungasaurus was the top predator, preying primarily on large sauropods such as Respiratory. It is also one of the very few dinosaurs for which scientists have found direct evidence of cannibalistic behavior. The story of Majungasaurus begins with the French paleontologist Charles Desperey, who in 1896 described the first theropod fossils from northwestern Madagascar. These included 2 teeth, a claw, and some vertebrae that had been collected along the Betsiboka River by a French army officer, and later deposited in the collection that would become part of the Université Claude Bernard Lyon 1. Desperey initially assigned these fossils to Megalosaurus, which at the time was a catch-all genus for any large theropod, and he named the species M. crenatissimus. The name comes from the Latin crenatus, meaning notched, and the suffix issimus, meaning most, referring to the many serrations along the edges of its teeth. Later, Desperey reassigned the species to the North American genus Dryptosaurus, another poorly understood dinosaur. Over the next century, French collectors recovered numerous fragmentary remains from Mahajanga province in northwestern Madagascar. Many of these were eventually housed in the Muséum National d'Histoire Naturelle in Paris. In 1955, René Lavocat described a theropod jawbone, or dentary, with teeth from the Maevarano Formation in the same region. These teeth matched those first described by Desperey, but the strongly curved jawbone was distinct from both Megalosaurus and Dryptosaurus. Using this dentary, Lavocat created the new genus Majungasaurus, combining an older spelling of the region Mahajanga with the Greek word seros, meaning lizard. In 1979, Hans-Dieter Suess and Philippe Taquet described a dome-shaped skull fragment, naming it Majungatholus atopus, which they believed belonged to a pachycephalosaur. This was the first report of a pachycephalosaur in the southern hemisphere. Major breakthroughs came in 1993, when scientists from the State University of New York at Stony Brook and the University of Antananarivo launched the Mahajanga Basin Project, a series of field expeditions to study late Cretaceous fossils near the village of Berivotra. Among these researchers was paleontologist David W. Krause. The first expedition uncovered hundreds of theropod teeth identical to those of Majungasaurus, including some attached to a small premaxilla, described in 1996. Subsequent expeditions produced tens of thousands of fossils, many belonging to species previously unknown, dramatically increasing the known diversity of the region's ancient life. In 1996, an exceptionally complete theropod skull was discovered, preserved in incredible detail. Atop this skull was the dome-shaped swelling previously attributed to Majungasaurus atopus. In 1998, this fragment was reclassified as belonging to an abellisaurid rather than a pachycephalosaur. Even though the name Majungasaurus crenatissimus was older than Majungasaurus atopus, the type dentary of Majungasaurus was initially considered too incomplete to confirm that it belonged to the same species as the skull. Over the following decade, paleontologists recovered multiple partial skulls, dozens of skeletons ranging from juveniles to adults, hundreds of isolated bones, and thousands of shed teeth. Together, these fossils account for nearly all parts of the skeleton, though the forelimbs, much of the pelvis, and the tip of the tail remain largely unknown. This research culminated in a comprehensive 2007 monograph, a collection of 7 scientific papers covering every aspect of Majungasaurus's biology. The papers, published in the Society of Vertebrate Paleontology Memoirs, were written in English with abstracts in Malagasy. In this volume, Lavacat's dentary was re-evaluated and confirmed as diagnostic for the species, officially replacing the name Majungasaurus with Majungasaurus. Additional significant specimens excavated in 2003 and 2005 are still being prepared for study, ensuring that Majungasaurus continues to reveal new secrets about its life and its world. Majungasaurus was a medium-sized meat-eating dinosaur that lived in what is now Madagascar toward the very end of the age of dinosaurs, around 70 to 66 million years ago. Most individuals were about 5.5 to 7 meters long, which is roughly 18 to 23 feet, and weighed between 750 to 1,100 kilograms, or 1,650 to 2,430 pounds. However, fossils from larger specimens hint that some adults grew even bigger, possibly rivaling the size of its South American cousin Carnotaurus, and could have exceeded 8 meters, or about 26 feet in length. The skull of Majungasaurus is unusually well preserved and studied compared to most other meat-eating dinosaurs, and it closely resembled the skulls of other members of its family, the abellisaurids. Like its relatives, its skull was short in length compared to its height, though not as extreme as Carnotaurus. Large skulls measured about 60 to 70 centimeters long, roughly 24 to 28 inches. The front of the upper jaw, called the premaxilla, was tall and gave the snout a very blunt appearance, a typical trait for abellisaurids. What made Majungasaurus stand out, however, was the unusual width of its skull. Abellisaurids are known for the rough sculpted texture of the outer surfaces of their skull bones, and Majungasaurus was no exception. On its nasal bones, these textures were taken to an extreme. The nasal bones were thick, fused together, and featured a low central ridge along the part closest to the nostrils. On top of the skull, a dome-shaped horn projected from the fused frontal bones, a distinctive feature of this dinosaur. In life, these bony structures were likely covered with some kind of skin or keratin, similar to the material found in modern bird beaks and horns. Advanced imaging techniques like CT scans reveal that both the nasal bones and the frontal horn had hollow cavities inside, possibly to make the skull lighter. The teeth of Majungasaurus were typical of abellisaurids, with short, blunt crowns, but it had more teeth than most relatives, 17 in the upper jaw and 17 in the lower jaw, second only to Rugops. The rest of the skeleton, the postcranial bones, closely resembled those of Carnotaurus and Orcosaurus, the only other abellisaurids for which complete skeletons have been found. Majungasaurus walked on 2 legs, using its long tail to balance its body and place its center of gravity over the hips. Its neck vertebrae, while full of holes and cavities to reduce weight, were robust and strong, with large muscle attachments and interlocking ribs, tendons attached to these ribs, giving the neck a forked appearance, and the long depressions along the sides of the cervical ribs further lightened the load without sacrificing strength. Altogether, these adaptations created a powerful and muscular neck. Its arms were short and curved, resembling those of Allosaurus and Carnotaurus. The forelimbs were tiny, with 4 reduced fingers, of which the outer fingers were very small and likely lacked claws. Unlike most dinosaurs, the hand bones of Majungasaurus were fused and immobile, making the hands essentially useless for grasping. A well-preserved specimen described in 2012 confirmed that the lower arms were short but strong, and the hands contained 4 small, probably inflexible fingers, with no claws. The legs of Majungasaurus were thick and sturdy, though short compared to its body. Its lower leg bone, the tibia, was even more robust than that of Carnotaurus, and the ankle bones were fused for extra strength. The feet had 3 main toes that bore the dinosaur's weight, while a smaller first toe did not touch the ground. Majungasaurus belongs to the theropod family Abelisauridae, which is grouped with the Noasauridae family in the superfamily Abelisauroidea. This superfamily is part of a larger group called Ceratosauria. Abelisaurids are recognized for their tall skulls with blunt snouts, rough sculpted facial bones, extremely small forelimbs, and stout hind limbs, among other unique features. Despite decades of study, the evolutionary relationships within the Abelisaurids remain confusing. Some studies suggest that Majungasaurus is closely related to the South American Carnotaurus, while others could not confidently place it. The most recent research, which uses the most complete fossil information, suggests that Majungasaurus is more closely related to Indian dinosaurs such as Rajasaurus and Indosaurus, while South American Abelisaurids like Carnotaurus, Allosaurus, Equirixinatosaurus, Ilocalesia, and Abelisaurus, as well as African rugops, belong to separate groups. This may indicate that Abelisaurids evolved into different lineages in the western and eastern parts of the ancient supercontinent Gondwana, highlighting the diversity and geographic spread of these remarkable predators. Majungasaurus is especially notable for the unusual decorations on its skull, which set it apart from many other dinosaurs. Its nasal bones were swollen and fused together, and it had a prominent horn on its forehead. While other ceratosaur dinosaurs like Carnotaurus, Rajasaurus, and Ceratosaurus itself also had crests or bumps on their heads, Majungasaurus's skull features were particularly distinctive. Scientists believe these head ornaments were likely used in interactions with other members of the same species, perhaps for showing dominance or signaling, although the exact way they were used remains uncertain. Interestingly, the hollow cavity inside the frontal horn would have made it weaker, suggesting it was probably not used in direct fights. Instead, the horn may have served mainly as a visual display. Although individual Majungasaurus varied in their skull decorations, there is no evidence that males and females had different ornamentation. The shape of Majungasaurus's skull and those of other Abelisaurids suggest that these dinosaurs hunted differently from most other theropods. Many theropods had long, low, and narrow skulls, which were very good at handling strong vertical forces from biting, but were less effective against twisting forces. Their hunting style may have been somewhat similar to modern dogs with long and narrow snouts, delivering multiple bites to gradually weaken their prey. In contrast, Abelisaurids, and especially Majungasaurus, had taller, wider, and often shorter skulls, designed for a different approach. With a short, broad snout, Majungasaurus may have used a strategy more like modern big cats, delivering a single powerful bite and holding on tightly until the prey was subdued. Among Abelisaurids, Majungasaurus had an exceptionally broad snout, and its entire body shows adaptations that support this bite-and-hold hunting style. Its neck was reinforced with strong vertebrae, interlocking ribs, ossified tendons, and enlarged areas for muscle attachment on both the vertebrae and the back of the skull. These features would have helped Majungasaurus keep its head steady, even while its prey struggled violently. The skull bones were further strengthened by minerals formed from the skin, giving them a rough texture that added durability. The nasal bones were thick and fused, providing extra strength for powerful bites. At the same time, the lower jaw had large openings on each side called fenestrae, and certain joints between the jaw bones allowed flexibility, although not nearly as much as a snake. These adaptations may have helped prevent the lower jaw from breaking while holding on to struggling prey. The front teeth of the upper jaw were thicker and stronger than the others, acting as anchor points for gripping. The overall low height of its teeth reduced the risk of breaking during a fight. Unlike the teeth of Allosaurus and most other theropods, which curved both forward and backward, Majungasaurus's teeth curved only on the front edge, while the back edge was straighter, likely helping to hold prey rather than slice it. Fascinatingly, studies show that Majungasaurus replaced its teeth extremely quickly, anywhere from 2 to 13 times faster than other theropods, with an entire set renewed in just 2 months. One reason for this rapid replacement may have been frequent gnawing on bones. Majungasaurus was the top predator in its environment, and the only large herbivores around were massive sauropods like Respiratory. Scientists think that Majungasaurus, along with other abelisaurids, may have specialised in hunting these enormous slow-moving herbivores. The reinforced skull and neck would have been very useful for a bite-and-hold attack against animals of such tremendous size and strength. Its legs were short and strong rather than long and slender like those of most other theropods, suggesting that it was built more for power than speed. These stocky, muscular legs lowered its centre of gravity, making it stable and formidable when grappling with giant prey. Although it probably could not run as fast as other theropods of similar size, Majungasaurus would have had no trouble keeping pace with the slow-moving sauropods it hunted. Evidence for this diet comes from Majungasaurus tooth marks found on Rapetosaurus bones, showing that it at least fed on these massive herbivores, whether it killed them itself or scavenged them. Although sauropods were likely the preferred prey of Majungasaurus, discoveries published in the year 2007 revealed a surprising twist. These fearsome predators also ate members of their own species. In Madagascar, paleontologists uncovered numerous Majungasaurus bones that bore tooth marks strikingly similar to those found on sauropod bones from the same sites. These marks were perfectly matched in spacing to the teeth of Majungasaurus jaws, were exactly the same size as its teeth, and even showed smaller notches consistent with the serrated edges of Majungasaurus teeth. Since Majungasaurus is the only large theropod known to have lived in this region, the simplest explanation is that these dinosaurs were feeding on each other. Earlier ideas that the Triassic dinosaur Calophysis engaged in cannibalism have since been disproven, making Majungasaurus the only non-bird theropod with confirmed cannibalistic behaviour, though there is some evidence suggesting that other species may have occasionally done the same. It remains unclear whether Majungasaurus actively hunted its own kind or merely scavenged the carcasses of others. However, researchers have pointed out that modern Komodo dragons sometimes kill each other while competing over a carcass and then eat the remains of their rival. This behaviour may hint at a similar strategy in Majungasaurus and perhaps other theropods, showing that cannibalism may have been part of survival strategies in these predatory dinosaurs. Majungasaurus's respiratory system has been reconstructed thanks to a remarkably preserved series of vertebrae, catalogued as UA 8678 from the Maevarano Formation. Many of these vertebrae, along with some ribs, contained hollow spaces known as pneumatic foramina, which likely formed as air sacs similar to those of modern birds invaded the bones. In birds, the neck vertebrae and ribs are hollowed by the cervical air sac, the upper back vertebrae by the lungs, and the lower back and hip vertebrae by the abdominal air sac. The presence of similar structures in Majungasaurus suggests it had a comparable system. These air sacs may have allowed a primitive form of what is called flow-through ventilation, where air moves through the lungs in one direction, preventing the mixing of oxygen-rich incoming air with carbon dioxide-laden exhaled air. Though complex, this type of breathing is highly efficient and may have supported the active predatory lifestyle of Majungasaurus. The discovery of these pneumatic spaces also carries significant evolutionary implications. The line of ceratosaur theropods, which includes Majungasaurus, diverged very early from the line of tetaneuran theropods, from which birds eventually evolved. The presence of an avian-like respiratory system in both groups suggests that such a system evolved long before birds appeared, reinforcing the idea that modern birds inherited their breathing adaptations directly from their dinosaur ancestors. Computed tomography, or CT scanning, of a complete Majungasaurus skull, catalogued as FMNH PR 2100, has provided additional insights. These scans allowed scientists to roughly reconstruct the dinosaur's brain and inner ear structure. Overall, Majungasaurus had a very small brain relative to its body size, similar to many other non-coelurosaurian theropods, and its overall brain shape was conservative, more comparable to modern crocodilians than to birds. One notable difference from other theropods was the size of the flocculus, a part of the cerebellum that coordinates eye and head movements. In Majungasaurus and other abelisaurids such as Indosaurus, this region was relatively small, suggesting that these dinosaurs did not rely on quick head movements when hunting prey. The inner ear of Majungasaurus also provides clues about its behaviour. The semicircular canals, which help with balance, indicate how the dinosaur held its head. When scientists adjusted the skull so that the lateral semicircular canal was parallel to the ground, the entire head was nearly horizontal. This is in contrast to many other theropods, whose heads tilted downward when alert. The lateral semicircular canal in Majungasaurus was also longer than in its more primitive relative Ceratosaurus, suggesting it could detect side-to-side head movements more acutely, perhaps aiding in hunting or environmental awareness. Pathologies, or signs of disease and injury, have also been documented in Majungasaurus bones. A report in 2007 examined at least 21 individuals and identified 4 with clear abnormalities. While pathologies had previously been studied in large tetaneuron theropods like Allosaurids and Tyrannosaurids, this was the first time an abelosauroid had been studied in such detail. Interestingly, no skulls showed bite marks, unlike Tyrannosaurids, which often display gruesome facial injuries. One specimen had a broken toe bone that had healed, indicating survival after injury. Most of the bone abnormalities were found in the vertebrae. A juvenile's dorsal vertebra showed a bony growth on its underside, likely resulting from cartilage or ligament turning into bone during development, though the exact cause was unknown. A small tail vertebra also had a growth on the top of its neural spine, similar to features seen in Allosaurus and Masiakasaurus, probably caused by ossification of ligaments along the spine. The most extreme pathology was found in a series of 5 large tail vertebrae. The first 2 vertebrae showed minor issues, including a large groove along the left side, but the next 3 were completely fused into a solid bony mass. No vertebrae were found after the 5th, indicating that the tail ended abruptly. From the size of the last vertebra, scientists estimated that about 10 tail vertebrae were lost. This could have been caused by severe trauma, followed by infection in the remaining vertebrae, or an infection that caused the tail tip to die and fall off. This represents the first known case of tail truncation in a non-bird theropod. The relatively small number of specimens showing pathologies suggests that injuries accumulated over the lifetimes of individual dinosaurs. Most well-preserved Majungasaurus show no signs of injury, while a few had multiple pathologies. This pattern mirrors that seen in other large non-bird theropods and may reflect a snowball effect. Once an individual was injured or infected, its weakened body became more susceptible to further injuries and ailments. Majungasaurus is exceptionally well understood because many fossils of this dinosaur, representing individuals of different ages, have been found in remarkably good condition. Scientists have been able to study how it grew and developed over time. Its skull, in particular, shows fascinating changes as it matured. The jugal, postorbital and quadradojugal bones of the skull became taller, thicker, and more robust, while the bones gradually fused together more tightly. At the same time, the eye sockets became relatively smaller as the dinosaur aged. These changes suggest that young Majungasaurus likely ate different kinds of food compared with fully grown adults, hinting at a shift in diet as the animal grew. Research led by Michael Demick and colleagues has revealed that Majungasaurus grew very slowly compared to many other theropod dinosaurs. By examining the growth rings in its bones, lines that mark periods when growth slowed or stopped, they found that it took nearly 20 years for Majungasaurus to reach full maturity. This unusually slow growth rate may have been influenced by the harsh and challenging environment in which it lived. Interestingly, other members of the Abelisaurid family, which includes Majungasaurus, also grew at comparably slow rates. While adult Majungasaurus dominated the landscape, younger individuals may have faced threats even from unexpected predators. Fossil evidence suggests that subadult Majungasaurus could have fallen prey to the giant prehistoric snake Matsoya, though taking on such large prey would have been risky for the snake, and potentially dangerous for it as well. All known fossils of Majungasaurus come from the Maivarano Formation in the Mahajanga province of northwestern Madagascar. Most of the well-preserved skeletons, including the most complete specimens, were discovered in a section of the formation called the Anembalemba member. However, teeth of Majungasaurus have also been recovered from the layers below, known as the Massarobe member, and from the layers above called the Miadana member. While these sediment layers have not yet been dated directly with radiometric techniques, scientists have used biostratigraphy, the study of the ages of fossils, and paleomagnetism to estimate their age. The evidence indicates that these rocks were deposited during the Maastrichtian stage of the late Cretaceous period, which lasted from 70 million to 66 million years ago, Fossilised teeth of Majungasaurus are found throughout the Maastrichtian, right up until the very end of the stage, when all non-avian dinosaurs went extinct. At the time Majungasaurus roamed, Madagascar was already an island, having separated from the Indian subcontinent less than 20 million years earlier. The island was moving northward, but was still positioned about 10 to 15 degrees further south than it is today. The climate was semi-arid, with clear seasonal changes in both temperature and rainfall. Majungasaurus lived on a coastal floodplain that was crisscrossed with sandy river channels. Geological studies indicate that during the early wet season, these channels often experienced debris flows, carrying sediment and burying the remains of animals that had died during the dry season. This process helped preserve the fossils so exceptionally well. Sea levels were also rising steadily during this time, a trend that continued into the Paleocene epoch. This means that Majungasaurus likely explored coastal areas, including tidal flats, while the nearby Berevotra Formation preserves evidence of the marine environments that existed alongside its habitat. The Maivarano Formation was home to a diverse and fascinating array of animals. Alongside Majungasaurus, fossils have been found of fish, frogs, lizards, snakes, 7 distinct species of crocodile relatives, 5 or 6 species of early mammals, the bird Verona, and several other birds. More unusual dinosaur species have also been discovered, such as the possibly flight-capable Dromaeosaurid rahonaves, the Noasaurid Masiakasaurus, and 2 large titanosaurian sauropods, including Rapetosaurus. Among all these creatures, Majungasaurus was by far the largest land-based predator and likely ruled as the top carnivore of its ecosystem. However, large crocodile-like predators, such as Majungasaurus and Trematochampsa, may have competed with it in areas near water, adding an extra layer of danger to the environment. Tyrannosaurus is a genus of enormous meat-eating dinosaurs known as theropods. Its most famous species, Tyrannosaurus rex, is often called T-Rex, or simply T-Rex. This species is one of the best known and most thoroughly studied theropods ever discovered. Tyrannosaurus lived across what is now Western North America, which during its time formed a separate island continent called Laramidia. Unlike other members of its family, the Tyrannosaurids, Tyrannosaurus had a remarkably wide range. Its fossils have been discovered in several different geological layers that date back to the late Maastrichtian age of the late Cretaceous period, around 69 to 66 million years ago. There are even some isolated fossils that suggest Tyrannosaurus might have appeared slightly earlier, during the Middle Campanian age. It was the last of the Tyrannosaurids and among the final non-bird dinosaurs to walk the Earth before the massive extinction event that ended the Cretaceous period. Like its tyrannosaur relatives, Tyrannosaurus was a bipedal predator, walking on 2 strong legs and possessing a huge skull balanced by a long, heavy tail. Its arms were short, especially when compared to its powerful hind limbs, yet they were unusually strong for their size and ended in 2 sharp claws. The most complete skeleton found measures about 12 meters and 3 decimeters to 12 meters and 4 decimeters in length, roughly 40 to 41 feet. However, modern research suggests that some individuals may have grown even larger, reaching over 13 meters or around 43 feet in length, standing 3 meters and 7 decimeters to 4 meters, about 12 to 13 feet tall at the hips, and weighing approximately 8 metric tons, or nearly 9 US short tons. Although a few other giant theropods might have matched or exceeded its size, Tyrannosaurus remains one of the largest land predators ever known. Its bite force is believed to have been the strongest of any land animal. In its environment, Tyrannosaurus rex was by far the largest meat-eater and almost certainly an apex predator. It hunted large plant-eating dinosaurs, including hadrosaurs, and likely targeted young or smaller armoured species like ceratopsians and ankylosaurs. Some evidence even suggests it could have taken on massive sauropods on occasion. For many years, scientists debated whether Tyrannosaurus was primarily a hunter or simply a scavenger. Today, most paleontologists agree that it was both, capable of hunting live prey while also scavenging when the opportunity arose. A number of T-Rex fossils are nearly complete, and at least one specimen has even shown traces of soft tissues and preserved proteins. The wealth of fossil material has allowed researchers to study many aspects of the dinosaur's life, including how it grew, how its body worked, and how it might have moved. Its feeding behaviour, metabolism, and speed continue to spark discussions among scientists. The classification of Tyrannosaurus is also debated. Its close Asian relative, Tarbosaurus bataar, is sometimes considered a species of Tyrannosaurus. Several North American tyrannosaur fossils have been merged with Tyrannosaurus, while some T-Rex specimens have been suggested as distinct species, such as the more recently described T. macriensis, though the validity of these names remains controversial. Tyrannosaurus has been one of the most famous dinosaurs since the early 20th century. Science writer Riley Black has even called it the ultimate dinosaur. Its fossils are popular museum exhibits, and the species has become a star in movies, documentaries and other media, most notably in Jurassic Park. The first fossil now recognised as belonging to Tyrannosaurus rex was a tooth found in July of 1874 on South Table Mountain in Colorado. It was discovered by a student named Peter T. Dotson under the guidance of geologist Arthur Lakes near the town of Golden. In the early 1890s, paleontologist John Bell Hatcher collected additional bones in eastern Wyoming. At the time, these fossils were thought to belong to a different large dinosaur, Ornithomimus grandis, now known as Dinodon, but today they are recognised as T-Rex remains. In 1892, Edward Drinker Cope discovered two large vertebrae and initially thought they belonged to a horned dinosaur, naming them Manospondylus gigas, which means giant porous vertebra, referring to the many holes in the bones. Later, in 1907, Hatcher identified these bones as those of a theropod, not a ceratopsid. By 1917, Henry Fairfield Osborn noticed similarities between Manospondylus gigas and Tyrannosaurus rex. However, because the bones were incomplete and one vertebra had been lost, Osborn did not merge the two names, instead labelling Manospondylus as uncertain. In June of 2000, the Black Hills Institute uncovered about 10% of a Tyrannosaurus skeleton, at a site possibly linked to the original Manospondylus discovery. The first partial skeleton of T-Rex was found by Barnum Brown, assistant curator at the American Museum of Natural History, in eastern Wyoming in 1900. Brown discovered another partial skeleton 2 years later in Montana's Hell Creek Formation, consisting of about 34 fossilized bones. Brown wrote at the time about the remarkable find, noting that the bones included the thigh bone, pelvic bones, upper arm bone, 3 vertebrae and 2 other unidentified bones of a giant carnivorous dinosaur unlike anything described before. In 1905, Osborn officially named this dinosaur Tyrannosaurus rex. The name combines the Greek words tyrannos for tyrant and sauros for lizard, while rex emphasizes its status as the king. The full name therefore means tyrant lizard king, highlighting its enormous size and dominance over other animals of its time. In the same year, Osborn also named another specimen Dynamasaurus imperiosus, but by 1906 he realized both skeletons belonged to the same species, and chose Tyrannosaurus as the official name. In 1941, the original T-Rex specimen was sold to the Carnegie Museum of Natural History in Pittsburgh, Pennsylvania, for $7,000. The original Dynamasaurus bones are now kept at the Natural History Museum in London. Interestingly, Dynamasaurus was later honoured in the name of a newly described tyrannosaurid, Dynamoptera dynastes, in 2018, inspired by Osborn's original naming. From the 1910s until the late 1950s, Barnum Brown's discoveries were the only known T-Rex specimens, as the Great Depression and World Wars kept many paleontologists out of the field. Starting in the 1960s, scientists and dinosaur enthusiasts renewed their fascination with Tyrannosaurus, sparking a wave of discoveries across western North America. Over time, 42 skeletons were recovered, ranging from small fragments to specimens that were 80% complete by bone count. One early find, in 1967, involved Dr. William McManus uncovering a skeleton known as Moore 008, which was only about 15% complete. Despite its incompleteness, the skull was carefully reconstructed and is now proudly displayed at the Museum of the Rockies. The 1990s brought an explosion of Tyrannosaurus discoveries, almost doubling the number of finds compared to all previous decades. Among these were 2 of the most famous and complete skeletons ever recovered, Sue and Stan. On August 12, 1990, Sue Hendrickson, an amateur paleontologist with remarkable dedication, uncovered the largest and most complete Tyrannosaurus skeleton known, with roughly 85% of its bones preserved, in the Hell Creek Formation. This incredible specimen was later named Sue in her honour. Sue became the centre of a heated legal battle over ownership, which was eventually resolved in 1997 in favour of Maurice Williams, the original owner of the land. Soon after, the Field Museum of Natural History purchased the fossil at auction for $7,600,000, setting a record as the most expensive dinosaur skeleton at the time, a title it held until Stan sold for $31,800,000 in 2020. From 1998 to 1999, museum staff devoted over 25,000 hours to carefully removing rock from the bones. The bones were then transported to New Jersey for mounting, before being returned to Chicago for final assembly. The fully mounted skeleton was unveiled to the public on May 17, 2000, at the Field Museum. Studies of Sue's bones revealed fascinating details about her life. She reached full size at 19 years old and lived until 28, which is the longest known lifespan of any Tyrannosaurus to date. Stan, the second most complete Tyrannosaurus skeleton, was discovered in 1990 in the Hell Creek Formation. Named in honour of amateur paleontologist Stan Sacrison, this specimen, catalogued as BHI 3033, had 190 bones recovered, representing about 70% of the total skeleton. Stan's bones revealed a life full of hardship, showing evidence of broken and healed ribs, a fractured and mended neck, and even a large hole in the back of the skull approximately the size of a Tyrannosaurus tooth. In 1998, a 20-year-old named Bucky Derflinger spotted a Tyrannosaurus toe sticking out of the ground, making him the youngest person ever to discover a T-Rex. The specimen, affectionately named Bucky, belonged to a young adult measuring about 3 meters tall and 11 meters long. Bucky is particularly remarkable as the first Tyrannosaurus to preserve a furcula, or wishbone, and it is now permanently displayed at the Children's Museum of Indianapolis. The early 2000s continued the trend of remarkable discoveries. In the summer of 2000, teams organised by the renowned paleontologist Jack Horner unearthed 5 Tyrannosaurus skeletons near the Fort Peck Reservoir. In 2001, the Burpee Museum of Natural History discovered a 50% complete skeleton of a juvenile Tyrannosaurus in the Hell Creek Formation. This young specimen, named Jane and catalogued as BMRP 2002.4.1, was initially believed to represent the first known pygmy tyrannosaurid, Nanotyrannus. However, further research clarified that Jane was a juvenile Tyrannosaurus, making her the most complete young T-Rex ever discovered. Jane is now exhibited at the Burpee Museum. In 2002, amateur collectors Dan Wells and Don Wyrick discovered another Tyrannosaurus skeleton, nicknamed Wyrex, which included 114 bones, representing 38% of the skeleton. The excavation, led by the Black Hills Institute, lasted 3 weeks in 2004 and was shared with the public through the first ever live online Tyrannosaurus dig, complete with daily updates, photos and videos. In 2006, Montana State University announced that it had obtained the largest Tyrannosaurus skull ever discovered, belonging to the MOR 008 specimen, measuring approximately 5 feet long. Later comparisons showed that the longest skull belonged to specimen LACM2, 3844 at 136.5 centimeters, while the widest skull, measuring 90.2 centimeters, belonged to Sue. Although skeletal remains are the most common evidence of Tyrannosaurus, rare footprints have also been attributed to this giant predator. The first footprint was discovered in 1983 at Philmont Scout Ranch in New Mexico by American geologist Charles Pillmore. Originally mistaken for the track of a hadrosaur, closer examination revealed a large heel not seen in ornithopod tracks, along with traces of a hallux, the dewclaw-like fourth toe of a tyrannosaur. This footprint was formally described in 1994 as Tyrannosauropus pilmerae by Martin Lockley and Adrian Hunt, making it likely the first known T-Rex track. The track measures 83 centimeters long and 71 centimeters wide and was made in what was once a vegetated wetland mudflat. A second possible Tyrannosaurus footprint was reported in 2007 by British paleontologist Phil Manning, discovered in the Hell Creek Formation of Montana. This track, measuring 72 centimeters in length, is slightly smaller than the earlier New Mexico track. Although it is not absolutely confirmed to belong to Tyrannosaurus, it remains the most plausible candidate, as this was the only large predator in the region at the time. In 2016, researchers Scott Persons, Phil Curry, and their team described a series of footprints from Glen Rock, Wyoming, dating to the Maastrichtian stage of the late Cretaceous, originating from the Lance Formation. These tracks were believed to have been made by either a juvenile Tyrannosaurus or the debated Nanotyrannus lancensis. Measurements and footprint spacing suggested that the dinosaur was walking at a speed between 2.8 and 5 miles per hour, with an estimated hip height of 1.56 to 2.06 meters. A follow-up study in 2017 revised the speed estimates, increasing them by 50 to 80%, showing that even the young or smaller tyrannosaurs could move with surprising agility. Tyrannosaurus rex was one of the largest land-dwelling meat-eating dinosaurs to ever walk the earth, a true giant among predators. Among the most complete and famous skeletons is one nicknamed Sue, which now resides in the Field Museum of Natural History in Chicago. Sue stretched an astonishing 12 meters and 30 to 12 meters and 40 centimeters long, which is roughly 40 to 41 feet. At the hips, she stood around 3 meters and 66 to 3 meters and 96 centimeters tall, equivalent to about 12 to 13 feet. Modern studies, using a variety of methods, estimate that her body mass reached approximately 8 metric tons and 400 to 8 metric tons and 46 kilograms, roughly 9 tons in the American short ton system. Another notable specimen, nicknamed Scotty at the Royal Saskatchewan Museum, may have been even longer, measuring an incredible 13 meters or 43 feet. Using a technique that estimates weight from the circumference of the thigh bone, scientists suggest Scotty may have weighed around 8 metric tons and 870 kilograms, making it the heaviest known T-Rex on record. Not every adult T-Rex grew to such monstrous sizes. Over the years, estimates of average adult weight have varied widely. Some scientists once suggested as little as 4 metric tons and 500 kilograms, or about 5 short tons, while others proposed weights exceeding 7 metric tons and 200 kilograms, almost 8 short tons. Today, most modern estimates place the typical adult between 5 metric tons and 400 kilograms and 8 metric tons, or roughly 6 to 9 short tons. A study in 2024 suggested that there was little difference in size between males and females, meaning that sexual dimorphism, differences between sexes, was probably minimal in T-Rex. The skull of Tyrannosaurus was another marvel of design. The largest known skulls measured up to 1 meter and 54 centimeters long, around 5 feet. Large openings in the skull, known as fenestrae, reduced weight without sacrificing strength, a common trait among all meat-eating theropods. Compared to other giant predatory dinosaurs, T-Rex's skull was unique. It was extremely wide at the back but had a narrow snout, giving it remarkably good binocular vision. Many skull bones were massive, and some, like the nasal bones, were fused to prevent movement, though many were filled with tiny air pockets, making them lighter. These adaptations, along with other features, allowed T-Rex to have an incredibly powerful bite, far stronger than any non-tyrannosaurid predator. The tip of its upper jaw was U-shaped, unlike the V-shaped jaws of most other large carnivores. This shape allowed it to tear away larger chunks of flesh and bone in a single bite, though it put more stress on the front teeth. T-Rex's teeth were fascinating in their variety and specialization. At the very front of the upper jaw were 4 teeth on each side, D-shaped in cross-section, closely packed, chisel-like, and curved backward. These features prevented the teeth from breaking when the dinosaur bit and pulled its prey. The remaining teeth were enormous and robust, more like deadly bananas than sharp daggers, spaced farther apart and reinforced with ridges. Adult T-Rex had about 12 upper teeth on each side, larger than those in the lower jaw except at the rear. The largest Rex tooth discovered so far, including its root, measured about 30 centimeters, or nearly 12 inches, making it the largest tooth of any meat-eating dinosaur ever found. The lower jaw was also strong and had 13 teeth at the front. Like many dinosaurs, both jaws contained numerous small holes in the bones, called foramina, which may have housed sensory nerves like in crocodiles, or supported structures such as scales or possibly lips, a theory supported by studies of tooth-wear patterns. Tyrannosaurus had a complex and powerful backbone. Its neck contained 10 vertebrae, the back had 13, and the sacrum had 5 fused vertebrae. The tail vertebrae are less certain but probably numbered at least 40. Sue's tail was displayed with 47 vertebrae. The neck naturally curved in an S shape like other theropods, but was exceptionally short deep and muscular to hold the enormous skull. The second vertebra, called the axis, was particularly short. Most of the neck vertebrae had a convex front and a concave rear, a shape that supported flexibility. They also contained single pluricoels, air-filled depressions that lightened the bones. The back vertebrae were strong but slimmed at the waist, with undersides that were keeled and large pluricoels. Their neural spines were roughened to attach powerful tendons. The sacral vertebrae were fused, lightened by air spaces, and connected to the pelvis with robust ribs and processes. The tail was long and heavy, balancing the massive head and torso, and providing room for enormous muscles that powered the legs. The 13th tail vertebra marked the transition from the deep base of the tail to a stiffer middle section. The underside of the torso was covered with 18 or 19 pairs of segmented belly ribs. The shoulder area of Tyrannosaurus was surprisingly long, longer than the entire arm. Its shoulder blade or scapula had a narrow middle section but flared out dramatically at the top, creating a broad surface for muscle attachment. At its front end, it extended into a long, forward-pointing projection that connected with the coracoid, a bone that was rounded in shape. Both shoulder blades were joined in the middle by a small wishbone-like bone called a furcula. The paired breastbones, or sternum, may have been made entirely of cartilage rather than solid bone. The arms of Tyrannosaurus were very short compared to the rest of its body. The upper arm bone, known as the humerus, was short yet sturdy, with a narrow upper section that ended in an unusually rounded head. The lower arm bones, the ulna and radius, were straight and much shorter than the humerus. Interestingly, the second hand bone, or metacarpal, was longer and wider than the first, which is the reverse of what is usually seen in other theropod dinosaurs. Tyrannosaurus's hands had only 2 fully functional clawed fingers, along with a tiny splint-like third metacarpal, which was a vestige of a third finger. The pelvis was massive, built to support powerful hind limbs. The top pelvic bone, called the ilium, was both very long and very high, providing a huge surface for muscles that moved the legs. The front pubic bone ended in a large boot-shaped expansion that was longer than the shaft of the bone itself. The rear ischium was slender and straight, pointing backward and slightly downward. Unlike the short arms, the hind limbs were enormous in proportion to the body, among the longest of any known theropod. The foot had a special feature called an arctometatarsus, meaning that the third metatarsal, the middle long bone in the foot, was pinched near the ankle. This third metatarsal was also unusually curvy. To balance the dinosaur's huge bulk, many bones throughout its skeleton were hollowed, reducing weight while maintaining strength. Tyrannosaurus is the defining genus of its broader family tree, serving as the standard for the superfamily Tyrannosauridae, the family Tyrannosauridae, and the subfamily Tyrannosaurinae. Other members of the same subfamily include the North American Daspletosaurus and the Asian Tarbosaurus, which are sometimes considered so similar to Tyrannosaurus that they have been treated as the same genus in certain studies. For a long time, tyrannosaurids were thought to descend from earlier giant theropods like megalosaurs and carnosaurs. More recent research, however, has placed them among generally smaller theropods called coelurosaurs. The earliest members of the tyrannosaur group were crested proceratosaurids, while later, more advanced tyrannosaur species belonged to a group called Pantyrannosauria. Tyrannosaurs started out as small meat-eating dinosaurs, but by the early Cretaceous, some had grown to enormous sizes. It seems likely that tyrannosauroids became dominant after other large theropods, such as allosauroids and megalosauroids, began to decline during the early stages of the late Cretaceous. Tyrannosauroids are identified by their fused nasal bones and unique teeth arrangement. Within Pantyrannosauria, species show special features in the hips, an enlarged hole in the quadrate bone, broad bones behind the eyes called postorbitals, and hourglass-shaped nasal bones. More advanced Pantyrannosaurs often lost the air-filled cavities in the nasal bones, had shorter arms relative to their legs, and some developed the arctometatarsalian foot. Eutyrannosaurs had rough nasal bones and a smaller external mandibular opening. Tyrannosaurids, including Tyrannosaurus, had solid skulls without movable joints or ornamental crests on the nose, and a distinctive lacrimal bone near the eye with a unique projection. Their skulls also featured an interfenestral strut, which was less than half the size of the maxillary fenestra. Many studies have shown that Tarbosaurus bataar is the closest relative, or sister species, of Tyrannosaurus rex. The discovery of the Tyrannosaurid Lithranax has reinforced this, suggesting that Tarbosaurus, Jucheng tyrannus, and Tyrannosaurus form a close-knit clade, with Lithranax as their sister group. A study in 2016 by Steve Broussard, Thomas Carr and colleagues even proposed that Tyrannosaurus may have migrated from Asia and could be descended from Tarbosaurus. In 1955, Soviet paleontologist Evgeny Maleev named a species Tyrannosaurus batar from Mongolia. By 1965, it was reclassified into its own genus, Tarbosaurus batar. While most paleontologists still treat the two as separate genera, some experts, including Thomas Holtz, Kenneth Carpenter, and Thomas Carr, argue they are similar enough to be considered the same genus. Several specimens from the late Cretaceous of China have been described as new Tyrannosaurus species, including T. lanpingensis from a tooth in the red beds of Yunnan in 1975, T. turpanensis from Xinjiang in 1978, and T. luanchuanensis from Henan province in 1979 to 1980. However, these species were later all considered junior synonyms of Tarbosaurus batar by Holtz in 2004, as they lacked detailed descriptions. A large finger bone labelled VGI, number 231/3, was assigned to Tyrannosaurus sp. by Yakov in 2000 from the lower Maastrichtian deposits in Bereslavka, Russia. Later, in 2004, Averyanov and Yakov suggested it might instead be part of a ceratosaur. In their 2023 overview, Averyanov and Lopatin treated this bone and a single tooth from the same site as belonging only to indeterminate theropods. In 2001, tyrannosaurid teeth and a foot bone from near Jucheng, China, were named Tyrannosaurus juchengensis. A nearby site later yielded a right upper jaw and left a lower jaw assigned to the new genus Jucheng tyrannus in 2011. T. juchengensis is likely synonymous with Jucheng tyrannus, though it is considered a dubious name because the specimen lacks clear distinguishing features below the level of Tyrannosaurini. In 2006, a fragmentary tyrannosaurid lacrimal bone, labelled CM 9401, from the Judith River Formation in Fergus County, Montana, was described as a possible Tyrannosaurus species. This isolated right lacrimal, originally found alongside the giant crocodile Dinosuchus rugosus, resembles T-Rex in size and shape, lacking the horns seen in earlier tyrannosaurids. Its size places it within the range of known T-Rex individuals, suggesting that large tyrannosaurids existed during the Campanian stage around 75 million years ago, much earlier than the usual age of Tyrannosaurus rex, which lived around 68 to 66 million years ago. In 2018, a study of tyrannosaurid teeth from the Two Medicine Formation noted a premaxillary tooth, YPM VPPU 023469, that strongly matched Sue, one of the most complete T-Rex specimens. The same study suggested that CM 9401 may also have come from the Two Medicine Formation, specifically the Flag Butte member, which dates to around 77 to 76.3 million years ago. This makes these specimens much older than any other known Tyrannosaurus, placing them alongside Daspletosaurus. In 2025, Charlie Shearer used these specimens to argue that Tyrannosaurini, the group including T-Rex, did not evolve directly from Daspletosaurus. In a study published in the year 2022, the paleontologist Gregory S. Paul, along with his colleagues, proposed a bold and controversial idea. The famous dinosaur Tyrannosaurus rex, as it has long been understood, might actually represent not just one species, but three distinct species. According to their research, the classic Tyrannosaurus rex remains the type species, while two additional species had gone unrecognized until now: Tyrannosaurus imperator, meaning tyrant lizard emperor, and Tyrannosaurus regina, meaning tyrant lizard queen. The holotype, or main reference specimen for T. imperator, is the well-known Sioux skeleton, displayed at the Field Museum of Natural History in Chicago, whereas the holotype for T. regina is Wankel rex. Paul and his team based this proposed division primarily on notable differences in the bones of various Tyrannosaurus specimens. In particular, they focused on variations in the femur, or thigh bone, and other skeletal elements, observing that some were far more robust while others were more slender. They also considered differences in the number of small, slender teeth at the front of the lower jaw, known as incisiform dentary teeth. According to their classification, the classic T-Rex was robust, with a moderately proportioned femur and a single slender tooth. T. imperator was also robust, but had a smaller femur length-to-width ratio and two slender teeth, and T. regina was more gracile, with a longer, thinner femur and a single slender tooth. Despite the apparent logic of this analysis, many leading paleontologists voiced skepticism. Experts such as Stephen Broussard, Thomas Carr, Thomas Holtz, David Hone, Jingmai O'Connor, and Lindsay Zano either criticised the study or expressed doubts when interviewed by the media. Their concerns were later formalised in a technical paper. Holtz and Zano remarked that while it was entirely possible more than one species of Tyrannosaurus existed, the evidence presented by Paul and his team was insufficient to definitively name the new species. Holtz even suggested that if Tyrannosaurus imperator was a separate species, it might actually represent the same species as Nanotyrannus lancensis, in which case it would need to be called Tyrannosaurus lancensis. O'Connor, curator at the Field Museum where Sue is displayed, considered the evidence too weak to justify changing exhibit signs. Broussard, Carr and O'Connor believed the proposed differences could simply reflect natural variation among individual T-Rex dinosaurs. Both Carr and O'Connor also pointed out that the study failed to determine which specimens belonged to which of the newly proposed species. Interestingly, Philip J. Curry, who had initially co-authored the study, later withdrew because he did not wish to be involved in naming new species. In a follow-up paper released in the year 2025, Paul defended his earlier conclusions, arguing that T. imperator and T. regina could be distinguished further by examining the shapes of their postorbital bosses, bony ridges located behind the eyes. Separately, in the year 2024, a team led by Dalman described the remains of a tyrannosaur discovered back in 1983 in New Mexico's Hall Lake Formation, which dates from the Campanian to the early Maastrichtian stage of the late Cretaceous. This fossil, housed at the New Mexico Museum of Natural History and Science, included parts of the skull such as the right postorbital, right squamosal, left palatine, and an incomplete maxilla, along with lower jaw bones, isolated teeth, and chevrons. Some of these bones were briefly mentioned in 1984 as belonging to T-Rex, and were formally described in 1986. In 1990, researchers Lehman and Carpenter suggested that New Mexico Museum of Natural History and Science might belong to a completely new genus of tyrannosaur, though Carr and Williamson disagreed in the year 2000. Later, Sullivan and Lucas in the year 2015 argued there was little evidence to firmly identify it as T-Rex, tentatively labelling it as CF, Tyrannosaurus sp, and proposed that it likely lived before the Lancian stage, meaning before 67 million years ago, based on its probable association with Allosaurus fossils. Dalman and colleagues, in 2024, formally named the specimen Tyrannosaurus macraensis, referencing the McRae group of rock layers where the Hall Lake Formation belongs. The fossils of T. macraensis were found in strata estimated to be slightly older than the typical age range of T-Rex, which lived at the very end of the Maastrichtian stage. Initially, these layers were dated between 72.7 and 70,900,000 years ago, placing them in the late Campanian to early Maastrichtian. However, later in the same year, a conference abstract suggested the layers might be younger than 69 million years plus or minus 400,000, and older than 66 million years, bringing T. macraensis much closer in age to T-Rex. T. macraensis was estimated to have reached around 12 meters, or 39 feet in length, comparable to an adult T-Rex. Differences lay mainly in the skull. The lower jaw of T. macraensis was proportionally longer, shallower, and had a less pronounced chin, indicating a slightly weaker bite. Its teeth were blunter and more compressed side to side, and the postorbital crests behind the eyes were less prominent. Its skeletal anatomy also showed similarities to other tyrannosaurs like Tarbosaurus and Juchengtyrannus. The validity of T. macraensis was questioned again in 2025. Morrison and colleagues highlighted uncertainties in the dating, noting that the presence of Allosaurus fossils, used as biostratigraphic markers, complicates estimates due to variable sedimentation rates. They also suggested that the morphology of T. macraensis might fall within the known range of variations seen in T-Rex. Despite these questions, the researchers concluded that the genus Tyrannosaurus likely originated in Laramidia, with ancestors possibly migrating from Asia. Later that same year, Voris and colleagues regarded T. macraensis as a junior synonym of T-Rex, agreeing that its supposed distinguishing features fell within the variation already known for T-Rex. Likewise, Thomas Carr, in his study of the T-Rex holotype skull, did not recognize T. macraensis as a valid species, noting that all proposed features could be observed in the broader collection of T-Rex specimens. For many years, paleontologists unearthed fossils in the same rock layers as Tyrannosaurus rex, and initially thought they belonged to different species. Among these were fossils named Orblicodon and Albertosaurus megagracilis, the latter of which was renamed Dinotyrannus megagracilis in 1995. Over time, however, scientists came to a near-universal agreement that these fossils were not separate species at all, but rather belonged to juvenile or young T-Rex individuals. There is, however, one intriguing exception: a small but nearly complete skull discovered in Montana, measuring about 60 centimeters, or roughly 2 feet in length. This skull, catalogued as CMNH 7541, was first identified in 1946 by Charles W. Gilmore, who classified it as a species of Gorgosaurus, calling it Gorgosaurus lancensis. More than 40 years later, in 1988, the skull was re-examined by the famous paleontologist Robert T. Bakker, together with Phil Curry and Michael Williams, who at the time was curator of paleontology at the Cleveland Museum of Natural History, where the skull was housed and still remains on display. Their study revealed that the bones in the skull were fully fused, which usually indicates that the animal had reached adulthood. Based on this observation, Bakker and his colleagues proposed a new genus, Nanotyrannus, meaning dwarf tyrant, highlighting its apparently small adult size. They estimated that this dinosaur would have measured about 5.2 meters, or roughly 17 feet, in length when it died. However, the debate was far from over. In 1999, Thomas Carr conducted a detailed analysis and determined that the skull actually belonged to a juvenile, not an adult. This led Carr and many other paleontologists to classify Nanotyrannus as simply a young T-Rex, rather than a separate species. Then, in 2001, another juvenile tyrannosaur was discovered, nicknamed Jane, and catalogued as BMRP 2002.4.1. This fossil belonged to the same species as the original Nanotyrannus skull, and reignited the discussion about whether Nanotyrannus was truly a separate species. At a conference held in 2005 at the Burpee Museum of Natural History, many paleontologists who had previously supported the validity of Nanotyrannus, including Curry and Williams, acknowledged that Jane provided strong evidence that Nanotyrannus was, in fact, just a juvenile T-Rex. Not everyone agreed, though. Peter Larson continued to argue that Nanotyrannus might be a distinct, closely related species. He pointed to certain features in the skull, such as having 2 more teeth in each jaw than T-Rex, proportionally larger hands with extra bones on the third finger, and differences in the wishbone of an undescribed specimen. Larson also suggested that another dinosaur called Stygivinator, usually considered a young T. Rex, might actually represent an even younger Nanotyrannus. Later research, however, showed that other tyrannosaurids, such as Gorgosaurus, also lost teeth as they grew, and the differences in tooth count could simply reflect natural variation among individuals rather than species differences. By 2013, Thomas Carr concluded that all the traits used to argue for Nanotyrannus as a separate species could be explained by individual growth patterns, changes during development, or distortions of the fossil bones. Some research continued to question this. In 2016, studies of limb proportions by Persons and Curry suggested that Nanotyrannus might have moved differently from T-Rex, hinting at a potential distinction. Yet, paleontologist Manabu Sakamoto noted that these conclusions could be affected by the small number of specimens studied, and differences in movement do not automatically mean a separate species. That same year, Joshua Schmerge and Bruce Rothschild argued in favour of Nanotyrannus being valid based on specific skull features, like a groove in the jaw that is absent in T-Rex but present in some other tyrannosaurs. However, Carr and colleagues pointed out that such features were common and variable as tyrannosaurs grew, making it unreliable as evidence for a separate species. A major study in 2020 by Holly Woodward and colleagues reinforced the idea that all fossils attributed to Nanotyrannus were immature and most likely belonged to T-Rex. Later that year, Carr published a comprehensive paper on T-Rex growth, showing that the CMNH 7541 skull fit within the expected variation seen in juveniles of the species. He estimated that this young dinosaur was under 13 years old and had a skull less than 80 centimeters, or roughly 31 inches, long. Carr also noted that among 44T, Rex specimens studied, there were no noticeable differences tied to sex or evolutionary lineage, and traits thought to distinguish species actually changed consistently with age. He concluded that all Nanotyrannus fossils represented a transitional stage in T-Rex growth, bridging the gap between the smallest juveniles and the subadults, and capturing the dramatic change from the shallow skulls of young dinosaurs to the deep, massive skulls of fully grown adults. The debate did not completely end. In 2024, Nick Longrich and Evan Thomas Saita re-examined Nanotyrannus fossils and suggested, based on certain differences in anatomy, growth, and evolutionary relationships, that it could be a distinct species, possibly outside the Tyrannosauridae family. Yet, in 2025, Voris and colleagues argued that the apparent differences noted by Longrich and Saita were likely due to immaturity and problematic data, supporting the long-standing view that Nanotyrannus represents juvenile T-Rex specimens rather than a separate dinosaur species. The discovery that many fossils once thought to belong to different species were actually juvenile Tyrannosaurus rex has given scientists an incredible opportunity to study how these giant predators grew and aged. By examining these younger specimens, researchers have been able to trace changes in T-Rex as it matured, estimate how long it lived, and understand the speed at which it gained size and strength. The smallest known T-Rex, nicknamed the Jordan theropod and catalogued as LACM 28,471, weighed only about 30 kilograms, which is roughly 66 pounds. On the other end of the scale, the largest known adults, such as the famous Sioux, labelled FMNH PR 2081, were gargantuan, likely weighing close to 5,650 kilograms, or about 12,460 pounds. By looking at the structure of the bones under a microscope, a process called histology, scientists determined that the Jordan theropod had lived only 2 years before it died. Sioux, meanwhile, was originally thought to be 28 years old, which at the time seemed almost the maximum age a T-Rex could reach. More recent studies, however, have refined this estimate, placing Sioux's age between 27 and 33 years. Another large specimen, Trix, catalogued as RGM 792,000, appears to have been at least 30 years old when it died. Histology has also helped researchers determine the ages of many other T-Rex fossils. By plotting the ages of specimens against their sizes on a graph, scientists can create growth curves that show how quickly these dinosaurs grew over time. The growth curve of T-Rex has an S-shaped pattern. Juveniles remained relatively small, under 1,800 kilograms, or about 4,000 pounds, until they were around 14 years old. At that point, their growth accelerated dramatically. During this intense growth spurt, a young T-Rex could gain an average of 600 kilograms, or roughly 1,300 pounds each year for about 4 years. By the time it reached 18, the growth curve began to level off again, indicating that the dinosaur's body had slowed its expansion. For instance, only 600 kilograms separated the 28-year-old Sioux from a 22-year-old Canadian T-Rex specimen known as RTMP 81.12.1. Research published in 2004 confirmed these patterns, showing that the rapid growth phase began to slow at around 16 years of age. A study by Hutchinson and colleagues in 2011 supported these findings, but suggested that T-Rex may have grown even faster than previously thought, estimating that during the exponential growth phase, some individuals could gain up to 1,790 kilograms in a single year. This higher estimate brought the dinosaur's actual growth rate closer to what would be expected for an animal of its massive size. The sudden slowing of growth at the end of this rapid phase likely marked physical maturity, a theory reinforced by the discovery of medullary tissue in the thigh bone of a 16 to 20-year-old T-Rex from Montana, known as Moore 1125, or B-Rex. Medullary tissue, which appears only in female birds during ovulation, indicated that B-Rex was of reproductive age. Further analysis suggested that B-Rex was about 18 years old at death. In 2016, Mary Higby Schweitzer, Lindsay Zano and their team confirmed that the soft tissue in B-Rex's femur was indeed medullary tissue, also proving that the specimen was female. This discovery has since opened the door to determining the sex of other dinosaur species, as the chemical signature of medullary tissue is unmistakable. Other tyrannosaurids appear to follow similar growth patterns, though with slower growth rates that reflect their smaller adult sizes. In 2020, A study by Woodward and colleagues published in Science Advances revealed that T-Rex had the remarkable ability to slow its growth in response to environmental challenges such as food shortages. By examining 2 juvenile specimens aged between 13 and 15 years, housed at the Burpee Museum in Illinois, the researchers concluded that the rate at which T-Rex matured depended heavily on the availability of resources. This adaptability allowed it to survive in environments where food supplies fluctuated dramatically from year to year, a trait that may have given it an advantage over other mid-sized predators that struggled in such harsh conditions. The study also supported the idea that the controversial genus Nanotyrannus was not a separate species, but rather juvenile T-Rex, based on the growth rings in the bones of the studied specimens. Interestingly, more than half of all known T-Rex specimens appear to have died within 6 years of reaching sexual maturity, a pattern seen in other tyrannosaurs as well as some large, long-lived birds and mammals today. These species typically had high infant mortality, followed by relatively low death rates during the juvenile stage. Mortality then increased again once they reached sexual maturity, likely due to the physical demands of reproduction. Some researchers suggest that juvenile T-Rex fossils are rare because these dinosaurs survived their early years in large numbers and did not die often enough to be preserved as fossils. Other factors may include gaps in the fossil record and collectors' preference for large, spectacular specimens. In a lecture in 2013, Thomas Holtz Jr. famously remarked that dinosaurs lived fast and died young, reproducing quickly, while mammals, in contrast, lived longer because they took more time to mature. Paleontologist Gregorius Paul echoed this sentiment, noting that T-Rex reached adulthood quickly and lived fast-paced, dangerous lives, which contributed to their relatively short lifespans. The discovery of feathered dinosaurs sparked lively debate among scientists about whether Tyrannosaurus might have had feathers, and if so, how much of its body they would have covered. In 2004, a remarkable find in China's early Cretaceous Yixian Formation revealed a small tyrannosauroid called Dilong paradoxus, with delicate hair-like filaments, which are now considered the earliest forms of feathers. At the time, however, larger tyrannosaur species appeared to have skin impressions showing only scales. This led researchers to suggest that bigger tyrannosaurids might have lost their insulating feathers as they grew, because their larger bodies retained heat more efficiently than smaller ones. This idea was challenged when, a few years later, a gigantic relative called Yutyrannus huali, also from the Yixian Formation, was discovered with large parts of its body covered in feathers. This showed that even enormous tyrannosaurids could have been feathered, casting doubt on the idea that feather loss was simply a matter of body size. Then, in 2017, a study reviewing all known tyrannosaur skin impressions, including those from a Tyrannosaurus specimen nicknamed Wyrex, found patches of mosaic-like scales on the tail, hips, and neck. Researchers concluded that if feathers were present on large Tyrannosaurus, they were probably limited to the top of the trunk rather than covering the whole body. Around 2016, a conference abstract proposed that theropods like Tyrannosaurus might have had lips covering their upper teeth, instead of the bare, exposed teeth seen in crocodiles. This idea was based on the observation that enamel, the outer layer of teeth, needs to stay hydrated, a problem crocodiles don't face because they live in water. Some critics disagreed, suggesting instead that tyrannosaurids had large, flat scales on their snouts, more like crocodiles. However, closer examination revealed that crocodiles actually have cracked, keratinized skin, not smooth flat scales. By comparing the textured surfaces of tyrannosaur snouts with modern lizards, researchers concluded that tyrannosaurids had scaly, lizard-like skin rather than crocodile-like skin. In 2023, Cullen and colleagues returned to the lip debate, noting that the arrangement of small openings on tyrannosaur jaws and faces resembled those of modern lizards like monitor lizards and marine iguanas more than crocodiles. Comparing the teeth of Daspletosaurus with those of American alligators, they observed that tyrannosaur teeth showed little wear, while crocodile teeth were worn on their outer surfaces. This suggested that lips in tyrannosaurids could have kept the teeth moist, protecting them from damage. They also found that the proportion of tooth height to skull length in theropods was much smaller than in certain lizards, implying that the teeth were not so large as to prevent coverage by lips when the mouth was closed. As more Tyrannosaurus skeletons were discovered, scientists noticed differences in body builds, revealing 2 distinct types, or morphs, similar to patterns seen in other theropods. One was more heavily built, called the robust morph, and the other was more slender, called the gracile morph. Early studies suggested that these differences might reflect sexual dimorphism, with the robust morph being female. Evidence cited included wider hips in robust specimens, presumably to allow eggs to pass through the pelvic area, and a smaller chevron bone on the first tail vertebra, which was thought to aid egg laying, similar to what was once believed in crocodiles. However, evidence for sexual dimorphism in Tyrannosaurus has weakened in recent years. A study in 2005 showed that earlier claims of sexual dimorphism in crocodiles were mistaken, undermining comparisons with Tyrannosaurus. Furthermore, the very robust specimen nicknamed Sue was found to have a full-sized first tail chevron, suggesting that tailbones could not reliably indicate sex. Differences between Tyrannosaurus individuals might instead reflect geographic variation or age, with more robust individuals possibly being older animals. Remarkably, only one Tyrannosaurus specimen has been conclusively identified as female. Examination of B. rex revealed preserved soft tissues inside some bones, including medullary tissue, a specialized tissue found in modern female birds used to supply calcium for eggshell formation. Since medullary tissue naturally occurs only in females during egg production, this strongly indicates that B. rex was female and may have died while producing eggs. Modern crocodiles, the closest living relatives of dinosaurs, do not produce this tissue, which reinforces the idea that medullary tissue is a shared trait between birds and theropod dinosaurs, highlighting their close evolutionary relationship. For a long time, Tyrannosaurus rex was imagined in a very peculiar way. Like many other two-legged dinosaurs, it was often shown as a kind of living tripod, with its massive body leaning at a sharp angle, about 45 degrees or less from vertical, and its long tail dragging along the ground, much like a kangaroo balancing itself. This strange idea can be traced back to the year 1865, when Joseph Leidy reconstructed Hadrosaurus, making it the first dinosaur ever depicted standing on two legs. The concept stuck, and in 1915, Henry Fairfield Osborn, who was the president of the American Museum of Natural History, displayed the first complete Tyrannosaurus skeleton standing fully upright in this same pose. That towering posture remained on display for 77 years until the mount was finally taken apart in 1992. By the year 1970, paleontologists had realized that this upright stance was completely unrealistic. No living creature could have maintained such a position without severe problems. The straight-legged head-forward stance would have caused major stress and potential dislocations in several crucial joints, including the hips and the connection between the skull and spine. Despite being wrong, the original American Museum mount inspired countless films, murals and illustrations, such as Rudolf Zallinger's famous mural, The Age of Reptiles, at Yale University's Peabody Museum of Natural History. It was not until the 1990s, with the release of films like Jurassic Park, that the public finally saw Tyrannosaurus depicted more accurately. Today, in museums, books, and films, T-Rex is shown with its massive body held nearly parallel to the ground, its tail stretched straight out behind it to act as a perfect counterbalance for its enormous head. Scientists believe that if Tyrannosaurus wanted to sit, it could have shifted its weight backward, resting on a broad expansion at the end of its pelvic bone known as the pubic boot. With its weight supported there, it may have been able to move its hind legs freely. To rise again, it might have used its tiny forelimbs for a little extra support, a concept humorously called Newman's push-up theory, though experts still debate it. In any case, T-Rex probably could stand up even if it fell, simply by placing its legs beneath its centre of gravity, while using the tail as a counterbalance. Evidence from healed stress fractures in the forelimb suggests that, although small, the arms were indeed used at times, suffering injuries just like the rest of the body. When Tyrannosaurus was first discovered, only the upper arm bone, or humerus, was known. For the mounted skeleton shown to the public in 1915, Osborn guessed the forelimbs were longer and three-fingered, resembling those of Allosaurus. Yet a year earlier in 1914, Lawrence Lamb had described the much shorter, two-fingered arms of Gorgosaurus, a close relative, suggesting T-Rex likely had similar forelimbs. This idea remained speculative until 1989, when the first complete T-Rex arms were discovered belonging to Emor 550, nicknamed the Wankel Rex. The famous Tyrannosaurus specimen Sue also has complete forelimbs. T-Rex's arms are tiny compared to its enormous body, measuring only about 1 meter or 3 feet 3 inches long. Because of their size, some scientists once thought they were useless, but the bones reveal large areas for muscle attachment, indicating remarkable strength. As early as 1906, Osborn speculated the arms might have been used to hold onto a mate during reproduction. In 1970, Newman proposed that the forelimbs helped the giant dinosaur push itself up from a prone position. Over the years, many other theories have emerged, though some seem unlikely. In 2022, Predation suggested that the shrinking of the arms was a secondary adaptation. As tyrannosaurids evolved massive, powerful skulls and jaws, their arms became smaller to avoid injury from bites, particularly during feeding with other members of their species. Another possible use for the forelimbs was to hold struggling prey, while the tyrannosaur's enormous jaws delivered the killing blow. Supporting this idea, biomechanical studies show that T-Rex's arm bones were extremely thick, built to withstand immense force. The biceps alone could lift about 199 kilograms, or 439 pounds, and when combined with other muscles like the brachialis, the elbow flexion would have been even stronger. The arm muscles of T-Rex were about 3.5 times more powerful than those of a human. While the range of motion was limited, 40 degrees at the shoulder and 45 degrees at the elbow, these constraints, combined with massive strength, suggest the arms were specialised for holding tight during a struggle. Kenneth Carpenter and Matt Smith, in the first detailed scientific description of T-Rex's arms, dismissed ideas that the limbs were useless or that the dinosaur was a strict scavenger. Some researchers, like Stephen M. Stanley, proposed that the arms were used as weapons, slashing prey with claws to make long, deep cuts. However, this theory was rejected by Predation, who argued that Stanley had overestimated both the size and mobility of the forelimbs. Today, the picture of Tyrannosaurus's tiny yet powerful arms is more nuanced. They were small but incredibly strong, built to grasp, stabilize, and perhaps even aid in feeding, while the true terror of T-Rex lay in its enormous skull, crushing jaws and unmatched predatory might. For a long time, scientists believed that Tyrannosaurus, like most other dinosaurs, was cold-blooded, similar to modern reptiles. This meant that they thought its body temperature depended on the surrounding environment rather than being internally regulated. However, in the late 1960s, during a period later called the Dinosaur Renaissance, researchers such as Robert T. Bakker and John Ostrom began to challenge this idea. They suggested that dinosaurs might have been more active and energetic than previously imagined. Specifically, Tyrannosaurus rex was proposed to have been warm-blooded, meaning it could generate and maintain its own body heat, which would have allowed for a highly active lifestyle. Since that time, many paleontologists have worked to understand how well T-Rex could control its internal temperature. Evidence from bone tissue studies shows that young T-Rex grew at rates comparable to modern mammals and birds. These high growth rates suggest that T-Rex may have had a high metabolism, supporting the idea of warm-bloodedness. Growth patterns indicate that, like mammals and birds, T-Rex grew rapidly during its youth, but slowed down as it matured, rather than continuing to grow indefinitely as many other animals do. Another way scientists have studied T-Rex metabolism is through oxygen isotopes found in fossilized bones. The ratio of certain isotopes can reveal the temperature at which the bones formed. In one remarkable specimen, the difference in temperature between bones from the torso and the lower leg was no more than 4 to 5 degrees Celsius or 7 to 9 degrees Fahrenheit. Paleontologist Rhys Barrick and geochemist William Showers interpreted this small temperature difference as evidence that T-Rex maintained a fairly constant internal body temperature, a condition known as homeothermy. They suggested that its metabolism was somewhere between that of cold-blooded reptiles and warm-blooded mammals. Some scientists have cautioned, however, that fossilization can alter these isotope ratios, meaning they might not perfectly reflect the original temperatures. Barrick and Showers defended their conclusions by showing similar results in another large theropod, Giganotosaurus, from a different continent and tens of millions of years earlier. Other dinosaur groups, such as ornithischians, also showed signs of homeothermy, whereas contemporary lizards from the same formations did not. In 2022, Veman and colleagues used a different method, analysing byproducts of metabolism called lipoxidation signals. They found that dinosaurs, including T-Rex, had metabolisms similar to modern birds and even higher than those of mammals, suggesting that warm-bloodedness might have been a common trait among all dinosaurs. Even if T-Rex could regulate its temperature, this does not automatically mean it was fully warm-blooded. Some researchers suggest that large dinosaurs could have maintained stable internal temperatures simply due to their massive size, a phenomenon known as gigantothermy, similar to some modern sea turtles. Features in the skull of T-Rex, such as small openings called dorsotemporal fenestrae, may also have helped it regulate heat, much like modern crocodiles. In March of 2005, Mary Higby Schweitzer of North Carolina State University and her team announced a stunning discovery: soft tissue inside the marrow cavity of a fossilized T-Rex leg bone. This particular fossil, known as Museum of the Rockies specimen 1125, or Moore 1125, had been excavated from the Hell Creek Formation. Remarkably, the bone had been broken for shipping and was not preserved in the usual way because Schweitzer hoped to test it for soft tissue. Inside, the researchers found flexible, branching blood vessels and fibrous, elastic bone tissue. They also observed tiny structures resembling blood cells, similar to those of modern ostriches. It is still unclear whether these tissues are original or the result of an unknown preservation process, so the researchers have been careful not to claim definitively that they are original. If they are, studying proteins in these tissues could provide indirect clues about the dinosaur's DNA, since each protein is made by a specific gene. Before this discovery, scientists largely assumed preserved tissue was impossible and therefore did not look for it. Since then, similar tissue-like structures have been found in 2 more tyrannosaurs and a hadrosaur. Studies of these tissues suggest that birds are closer relatives to tyrannosaurs than any other modern animals. Chemical analysis of MOR 1125 also showed elements like sulfur, calcium and zinc, which are associated with bone remodeling, and indicated that the bone was similar in chemistry to that of living birds. In April of 2007, further studies reported in the journal Science showed that traces of collagen proteins from T-Rex bones closely matched those in modern chickens, followed by frogs and newts. Discovering proteins in creatures tens of millions of years old challenged the long-standing belief that fossilization completely replaced living tissues with minerals. These findings suggested that paleontology could expand into the field of molecular biology. Paleontologist Hans Larsen of McGill University called the discovery a milestone, noting that it could bring dinosaurs into the modern study of biology. Not everyone agreed, however. In 2008, Thomas Kay and colleagues argued that what Schweitzer had identified as soft tissue was actually biofilm created by bacteria, not preserved dinosaur tissue. They found tiny mineral spheres containing iron, which had been mistaken for blood cells, in various fossils, including ammonites, where no blood could have existed. Schweitzer strongly disagreed, noting that biofilms cannot create the branching, hollow tubes she observed. In 2011, Schweitzer and colleagues analysed the recovered collagen and found that the inner parts of the collagen coil were preserved, as would be expected after millions of years. Additional research has continued to confirm the presence of branching, vessel-like structures inside fossilised dinosaur bones. Scientists have long debated just how fast Tyrannosaurus rex could move. Estimates for its top running speed vary widely. Most calculations suggest it could reach speeds of around 9 meters per second, which is about 32 kilometers per hour, or 20 miles per hour. Some studies have proposed much slower speeds, between 4.5 to 6 and 8 meters per second, roughly 16 to 24 kilometers per hour, or 10 to 15 miles per hour. On the other hand, a few extreme estimates suggest Tyrannosaurus could theoretically sprint as fast as 20 meters per second, which is 72 kilometers per hour, or 45 miles per hour, though scientists agree that achieving such a speed is highly unlikely. Unlike more lightly built theropods such as Carnotaurus or Giganotosaurus, Tyrannosaurus was a massive, heavy predator, built more for power than for sprinting. Because no fossilized tracks show large theropods actually running, scientists have had to rely on various estimation techniques. In 2002, a team used a mathematical model, tested and validated on modern animals like alligators, chickens and humans, as well as on other species including emus and ostriches, to calculate the amount of leg muscle mass necessary for fast running, over 40 kilometers per hour or 25 miles per hour. Some researchers argue that Tyrannosaurus may have been able to move faster than previously thought because its hollow bones and other lightweight adaptations might have kept its adult weight down to around 4.5 metric tons, or roughly 5 short tons. They also note that modern animals such as ostriches and horses, which have long and flexible legs, can reach high speeds not necessarily through rapid strides, but through slower, longer steps. However, even moderately fast speeds for Tyrannosaurus would have required enormous leg muscles. To reach speeds over 40 km per hour, its leg muscles would have had to make up between 40 and 86% of its total body mass, a physically improbable scenario. With more typical muscle proportions, it likely could only walk or jog at about 18 km per hour, or 11 miles per hour. Paleontologist Thomas Holtz noted that tyrannosaurids, including Tyrannosaurus, and their close relatives had unusually long lower leg segments, the shin, foot and toes, compared to the upper leg. Their foot bones were tightly interlocked, forming a structure called an arctometatarsus, where the middle metatarsal bone is pinched between the two adjacent ones. This arrangement may have made running more efficient, allowing forces from the foot to transfer more effectively through the leg than in earlier theropods. Recent studies have also highlighted Tyrannosaurus's remarkable walking efficiency. In 2020, researchers led by Desecchi and colleagues examined over 70 species of theropod dinosaurs, comparing their leg proportions, body weights, and walking patterns. They estimated top running speeds as well as energy costs for relaxed movement like walking. They found that smaller theropods, such as the dromaeosaurids, had long legs adapted for speed, useful for hunting or escaping predators. For larger theropods over 1,000 kilograms or about 2,200 pounds, longer legs did not increase top running speed due to their massive size. Instead, long legs helped reduce energy costs while walking, improving foraging efficiency. Tyrannosaurs, including Tyrannosaurus itself, likely benefited from this adaptation, allowing them to cover long distances efficiently with less energy, hunt or scavenge more effectively, and need fewer hunting trips overall. Combined with evidence of agility, these adaptations suggest Tyrannosaurus may have used a stalking approach, quietly following prey over long distances before unleashing a sudden burst of speed to strike, similar in some ways to modern wolves, and possibly even hunting in groups at times. A study in 2021 by Pashavan Bailat and colleagues focused on Tyrannosaurus's preferred walking speed, finding it to be about 1.28 meters per second, roughly 4.6 kilometers per hour, or just under 3 miles per hour. Like modern animals, Tyrannosaurus likely reduced energy costs by walking in a rhythm that allowed its body to resonate naturally. Importantly, this study also considered the dinosaur's tail, which previous research had often ignored. The tail would have gently swayed with each step, aided by strong ligaments acting like rubber bands to store and release energy. Using a three-dimensional model of the Tyrannosaurus specimen known as Trix, scientists recreated the tail's motion, showing that this rhythm would have made walking remarkably energy efficient, much like the movement of humans, ostriches or giraffes today. Earlier studies have offered additional insights. A 2017 report estimated Tyrannosaurus's top speed at 17 miles per hour, or 27 kilometers per hour, but suggested that it likely exhausted its energy before reaching this speed, showing a curved relationship between size and speed. Another study that same year concluded that a fully grown Tyrannosaurus, weighing around 7 tons, would have been unable to run at all, as speeds above 11 miles per hour, or 18 kilometers per hour, could have broken its leg bones. This implies that other giant theropods like Giganotosaurus, Mappisaurus and Acrocanthosaurus may have faced similar limits. Yet research from 2019 by Eric Snively and colleagues painted a more nuanced picture. They found that Tyrannosaurus and other tyrannosaurids were more maneuverable than other large theropods due to their low rotational inertia and strong leg muscles. This would have allowed the dinosaur to make quick turns, pivot sharply on a planted foot while swinging the other leg, or even pirouette during a chase. Such agility likely contributed significantly to the evolutionary success of tyrannosaurids, combining a careful, energy-efficient stalking strategy with the ability to strike swiftly and decisively at prey. In the deserts and badlands of New Mexico and Wyoming, a series of rare fossil footprints and trackways have been discovered that reveal the presence of mighty tyrannosaurs roaming those lands millions of years ago. These tracks, classified under the ichnogenus Tyrannosauropus, are believed to have been made by Tyrannosaurus rex, based on the age of the surrounding rocks. The very first of these fossilized footprints was discovered in 1994 and was described by paleontologists Lockley and Hunt. It consisted of a single enormous footprint, a silent testament to the massive creature that had once thundered across the landscape. Later, in 2020, another remarkable pair of fossils was described, capturing a rare and astonishing moment. A large tyrannosaur pushing itself up from a lying position using its elbows in combination with the pads of its feet to rise to its full height. These extraordinary fossils were uncovered in Ludlow, Colorado and Cimarron, New Mexico, providing an almost cinematic snapshot of tyrannosaur behavior. In addition, another trackway discovered in 2018 in the Lance Formation of Wyoming may have been made by a juvenile Tyrannosaurus, or perhaps the controversial genus Nanotyrannus. The fossilized footprints not only reveal the presence of these predators, but also offer a glimpse into their walking speed. Estimates suggest that the creature that made this particular trackway was moving at a pace of 4.5 to 8 kilometers per hour, roughly 2.5 to 5 miles per hour, much faster than earlier assumptions about tyrannosaur walking speeds. Beyond their footprints, the senses of Tyrannosaurus rex have been revealed to be extraordinary. A study by Lawrence Whitmer and Ryan Ridgely of Ohio University showed that Tyrannosaurus had heightened sensory abilities similar to other coelurosaurs. Its eyes and head could move quickly and in a highly coordinated fashion, allowing it to track moving prey with precision. It could also detect low-frequency sounds over long distances, giving it the ability to sense the movements of prey or the calls of other animals from afar. On top of that, it had an exceptionally keen sense of smell, surpassing most other dinosaurs. In an independent study, Kent Stevens applied modified perimetry to facial reconstructions of Tyrannosaurus and discovered that it had a binocular vision range of 55 degrees, wider than that of modern hawks. Stevens concluded that Tyrannosaurus had 13 times the visual sharpness of a human, outstripping even an eagle, which is about 3.5 times sharper than a person. The farthest distance at which it could distinguish objects from the horizon may have been as much as 6 kilometers, or 3.5 miles, far beyond the 1-mile range of human vision. The famed paleontologist Thomas Holtz Jr. noted that such precise depth perception would have been vital for hunting. Tyrannosaurus faced prey like ceratopsians, including Triceratops, heavily armoured ankylosaurs such as Ankylosaurus, and swift hadrosaurs. For such dangerous or agile prey, every strike had to count, and sharp vision allowed the predator to get in, land that blow, and take the prey down with deadly efficiency. By contrast, predators like Acrocanthosaurus, which hunted slower-moving giant sauropods, had less need for precise depth perception. Even without direct evidence of the eye structures in Tyrannosaurus, scientists like Kenneth Carpenter estimated the size of its sclerotic ring based on comparisons with Gorgosaurus. Carpenter suggested that the ring of the famous Stan specimen measured about 7 centimeters across, with an internal opening of about 3.5 centimeters. From this, the estimated pupil diameter was roughly 2.5 centimeters, while the eyeball itself measured between 11 and 12 centimeters. Its eyeball depth was likely between 7.75 and 9.75 centimeters. Calculations of the eye's optical qualities indicate that Stan's Tyrannosaurus had a daytime vision specialist's eye, not particularly adapted for seeing in low-light conditions, suggesting that it hunted mainly during daylight. The olfactory senses of Tyrannosaurus were equally impressive. Its large olfactory bulbs and nerves, enormous compared to its brain size, point to a remarkable ability to smell. This would have allowed the Tyrannosaur to detect carcasses across vast distances, a skill comparable to modern vultures. In fact, studies of 21 non-avian dinosaur species show that Tyrannosaurus had the most highly developed sense of smell among them. Its hearing too was specialized, particularly in low-frequency sounds, indicated by the unusually long cochlea. These low sounds likely played an important role in communication, hunting, and social behavior. Remarkably, the snout of Tyrannosaurus appears to have been highly sensitive. A 2017 study of a related species, Daspletosaurus, showed that small openings in the facial bones contained sensory neurons, suggesting that tyrannosaurs may have used their snouts to carefully measure nest temperature, handle eggs, or gently care for hatchlings, similar to modern crocodilians. Further research in 2021 revealed that the neurovascular canals in the jaws of Tyrannosaurus allowed it to feel subtle differences in material and movement, aiding it in feeding on different parts of prey with precision. These sensitive canals may also have supported careful behaviors like nest building, parental care, and social communication with other tyrannosaurs. While some studies suggest that the network of facial nerves in Tyrannosaurus may not have been as extreme as in aquatic or semi-aquatic dinosaurs, it still indicates a highly functional face capable of nuanced sensory input, perhaps also serving roles in thermoregulation or signalling to other members of its species. Paleontologist Jack Horner has been one of the most vocal supporters of the idea that Tyrannosaurus was not a hunter at all, but rather a scavenger that survived by feeding exclusively on the remains of animals it did not kill itself. In popular books and articles, he has presented several arguments to back up this pure scavenger idea. One of the points he emphasizes is the unusual shortness of Tyrannosaurus's arms. Compared with other known predators, its forelimbs were tiny, and Horner argues that they would not have been strong enough to grab and hold struggling prey. Other scientists, however, such as Thomas Holtz Jr., have pointed out that many modern predators survive without using their front limbs to hunt. Animals like wolves, hyenas, and even secretary birds catch and kill prey without relying on their arms, and some extinct predators, such as the towering flightless phorusrhacids, would have similarly hunted without their forelimbs. Another key argument for scavenging comes from Tyrannosaurus's incredible sense of smell. Its olfactory bulbs and nerves were unusually large for its brain size, suggesting that it could detect scents over vast distances. Modern vultures, which can locate carcasses from miles away, provide a useful comparison. In studies of dinosaur olfactory bulbs, Tyrannosaurus had the most highly developed sense of smell of 21 dinosaurs examined, highlighting its potential skill at sniffing out carrion. The structure of Tyrannosaurus's teeth also supports the scavenger idea. Its massive teeth could crush bone, allowing it to extract as much nourishment as possible from the leftover scraps of a carcass, including the marrow inside bones, which is usually one of the few nutritious parts left after death. Fossilized droppings attributed to tyrannosaurs show fragments of bone, confirming that they did consume bones, although their teeth were not as specialized for systematically chewing bone as hyenas' teeth are. Some researchers argue that Tyrannosaurus's movement supports scavenging behavior. Because some of its potential prey could move quickly, the fact that Tyrannosaurus may have walked rather than run could indicate that it relied on scavenging. On the other hand, newer studies suggest that, while not as fast as today's large land predators, Tyrannosaurus may still have been quick enough to chase and hunt big hadrosaurs and ceratopsians. Evidence also exists for active hunting behavior. The positioning of Tyrannosaurus's eyes allowed for forward-facing vision, giving it binocular sight that was even slightly better than that of modern hawks. Such depth perception is generally found in predators, and it is hard to see why natural selection would have favoured this feature if Tyrannosaurus fed only on carcasses. Fossils tell even more dramatic stories. One skeleton of the hadrosaur Edmontosaurus annectans, discovered in what is now Montana, shows healed bite marks on its tail vertebrae inflicted by a Tyrannosaurus. The healing suggests that this Edmontosaurus survived an attack, meaning it was alive when bitten, clear evidence of attempted predation. There is some debate though. Studies from 2014 and 2020 propose that the tail injuries might have come from other causes, such as Edmontosaurus individuals stepping on each other or stress from movement, rather than a Tyrannosaurus attack. Additional evidence comes from encounters with Triceratops. Some Triceratops fossils show healed Tyrannosaurus tooth marks on a brow horn and parts of the neck frill. The horn was broken and then healed, showing the animal survived the encounter. In such confrontations, a Triceratops could have successfully defended itself, potentially wounding the Tyrannosaurus with its sharp horns. Fossils of the famous Tyrannosaurus specimen Sue tell similar tales. Sue shows a broken and healed fibula, damaged tail vertebrae, scars on facial bones, and even a tooth from another Tyrannosaurus embedded in a neck vertebra, all pointing to aggressive encounters with other animals of its kind. Fossil evidence from juvenile Tyrannosaurus also indicates that younger individuals could puncture bones in the same way adults did, showing that hunting and feeding techniques were practiced from a young age. Some scientists, including William Abler in 1992, have suggested that Tyrannosaurus may have had deadly bacteria in its mouth, much like the Komodo dragon, which could have infected prey with a single bite. Abler noticed that the tiny serrations on Tyrannosaurus's teeth formed little chambers, which might have trapped bits of flesh carrying bacteria. This theory was challenged by Horner and Don Lessem in 1993, who argued that the tooth serrations of Tyrannosaurus were more cube-like than the rounded serrations found on a Komodo dragon, casting doubt on the infectious bite idea. Tyrannosaurus and most other large theropods likely processed carcasses by shaking their heads from side to side, rather than chewing in a sophisticated manner. Its neck bones were flat at the joints, limiting maneuverability, unlike the more flexible skulls of other giant predators like Allosauroids. This method of feeding, similar to modern crocodiles, allowed Tyrannosaurus to tear flesh efficiently, whether scavenging or hunting. There is compelling evidence that tyrannosaurs, including the mighty Tyrannosaurus, sometimes turned on their own kind and practiced cannibalism. In fact, some of the clearest signs come from a Tyrannosaurus specimen whose bones, specifically the foot bones, humerus and metatarsals, bear unmistakable tooth marks made by another member of its species. These marks suggest that the animal was scavenging on a carcass, feeding on parts of a body where the meat was already mostly gone. Similar signs of cannibalism have been found in fossils from the Fruitland Formation and the Kirtland Formation, both dating to the Campanian stage of the late Cretaceous, as well as from the Ojo Alamo Formation, which is from the Maastrichtian stage. These discoveries hint that cannibalistic behaviour was not limited to Tyrannosaurus alone, but may have been common across several tyrannosaurid species living in what is now the San Juan Basin. Studies by paleontologists such as Curry, Horner, Erickson, and Longrich, published in 2010, support this view. They carefully examined Tyrannosaurus bones with tooth marks and concluded that these marks were likely the result of opportunistic scavenging rather than aggressive fights with other Tyrannosaurus individuals. After all, biting a rival's feet during a confrontation would have been difficult, making it more plausible that these marks were left on a dead body. While direct evidence of Tyrannosaurus raising its young is lacking, juvenile fossils and nests are extremely rare. Scientists have speculated that, like their closest living relatives, birds and crocodiles, these giant predators might have cared for and fed their offspring. Crocodiles and birds are often used as modern analogues for dinosaur parenting, and evidence of such behaviour has been observed in other dinosaurs. For example, Maiasaura peoplesaurum is famous for being the first dinosaur shown to raise its young, and some closely related oviraptorids suggest that theropod dinosaurs may have exhibited parental care as well. In 2001, Bruce Rothschild and his colleagues published a study investigating stress fractures and tendon injuries in theropod dinosaurs, exploring what these injuries might reveal about their behaviour. Stress fractures, caused by repeated strain rather than a single accident, indicate frequent physically demanding activities. Among 81 Tyrannosaurus foot bones studied, one showed a stress fracture, while none of the 10 hand bones examined had such injuries. Tendon injuries were found only in Tyrannosaurus and Allosaurus. One notable injury was observed on the humerus of Sue, the famous T-Rex specimen, at the origin of the deltoid or teres major muscles, suggesting a violent struggle with prey. The concentration of tendon injuries in the forelimbs and shoulders implies that these dinosaurs had a complex musculature, different from modern birds. Overall, the presence of stress fractures and tendon injuries points to a highly active, predation-based lifestyle, rather than a diet limited to scavenging. A separate study in 2009 found smooth-edged holes in the skulls of several Tyrannosaurus specimens including Sue and Emor 980, nicknamed Pex Rex. Initially, these holes were thought to result from bacterial infections or intraspecific combat, but researchers suggested that they might have been caused by parasites similar to Trichomonas, which commonly infect modern birds. These infections could have made feeding difficult, potentially leading to starvation in severely affected individuals. Later research, however, raised doubts about this theory. The bone changes in the T-Rex skulls did not perfectly match those caused by Trichomonas in birds, and the severity and speed of the disease in birds does not seem directly comparable to what might have occurred in Tyrannosaurus. Some scientists now believe that the lesions might instead have been caused by combat with other Tyrannosaurus, rather than disease. Altogether, the evidence paints a fascinating picture. Tyrannosaurus was a powerful predator, capable of hunting and wrestling prey, but also an opportunistic scavenger that did not shy away from feeding on members of its own species when the chance arose. Other tyrannosaurids may have shared this grim but practical habit, demonstrating the complex and sometimes ruthless nature of these prehistoric giants. Tyrannosaurus, the iconic giant predator of the late Cretaceous, roamed the lands during what scientists call the Lancian faunal stage, which corresponds to the Maastrichtian age, at the very end of the Cretaceous period. Its range stretched across what is now North America, from the northern regions of present-day Canada all the way down to at least New Mexico in the south, covering a vast portion of the western landmass known as Larimedia. Many of the most famous Tyrannosaurus fossils have been unearthed in the Hell Creek Formation, a remarkable sedimentary layer that preserves the ancient ecosystems of that time. During the Maastrichtian Age, this region was a warm, humid, subtropical landscape, teeming with life. The plant life was dominated by flowering plants, but forests of dawn redwoods or metasequoia also flourished, painting the landscape with towering greenery. The Hell Creek Formation has yielded an incredible variety of dinosaurs and other prehistoric creatures, showing just how rich and complex this ecosystem was. Among the dinosaurs that shared this habitat with Tyrannosaurus were the horned ceratopsians, such as Leptoceratops, Torosaurus, and the famous Triceratops, the duck-billed hadrosaurid Edmontosaurus annectens, the smaller lightly built Thescelosaurus, the armored ankylosaurs Ankylosaurus and Denversaurus, and the dome-headed pachycephalosaurs Pachycephalosaurus and Spherotholus. Fierce predatory relatives included the Paravian theropods Acaroraptor and Pectinodon, while other Maniraptoran theropods like Anzu, Ionophron, Trirarchuncus, and some indeterminate ornithomimids prowled the same lands. Even the skies were ruled by prehistoric flyers such as the pterosaur Infernodricon. Another key site for Tyrannosaurus fossils is the Lance Formation in present-day Wyoming. This formation has been interpreted as a bayou-like environment, similar to the swamps and marshes along today's Gulf Coast. Its dinosaurian inhabitants were very similar to those of Hell Creek, though the speedy Struthiomimus replaced its relative Ornithomimus, and the small ceratopsian Leptoceratops also thrived in this watery habitat. Moving further south, fossils from the Northhorn Formation in Utah show that Tyrannosaurus rex shared the landscape with titanic sauropods such as Eutetitan, ceratopsids like Torosaurus, and various hadrosaurids and troodontids whose exact identities remain unclear. In New Mexico, the McRae group has revealed the species Tyrannosaurus macraensis, living alongside the ceratopsid Sierraceratops, and possibly the enormous Titanosaur Alamosaurus. In Texas, fossils tentatively identified as Tyrannosaurus have been found in the Javelina Formation, an area that also hosted massive titanosaurs, either Alamosaurus or Eutetitan, the ceratopsid Bravoceratops, flying giants like Quetzalcoatlus and Wellenhopterus, and likely other troodontids and hadrosaurids. Scientists believe that the southern portion of Tyrannosaurus's range consisted mostly of semi-arid inland plains, which formed as the western interior seaway gradually retreated due to falling global sea levels. Evidence also suggests that Tyrannosaurus may have ventured into what is now Mexico, particularly the Lomas Colorados Formation in Sonora. While no complete skeletons have been discovered there, 6 shed and broken teeth have been carefully compared to those of known Tyrannosaurus specimens and appear to match perfectly. If these teeth truly belong to Tyrannosaurus, it would mean that the species' range extended farther south than previously imagined. Some scientists even propose that tyrannosaurs may have originated in Asia, migrating to North America sometime before the end of the Cretaceous. Modern studies offer fascinating insights into the population of these colossal predators. A 2021 study led by Charles Marshall and colleagues estimated that the total number of adult Tyrannosaurus at any given time may have been around 20,000 individuals. Computer models suggest that this number could not have been lower than 1,300, nor higher than 328,000. Marshall and his team also noted that 20,000 might be an underestimate, especially since a population that's small would be vulnerable to disease outbreaks. Over the entire existence of the genus, scientists estimate there were about 127,000 generations, adding up to roughly 2.5 billion Tyrannosaurus that ever lived until their extinction. According to the same study, in a population of 20,000 adults, a region the size of California could have supported as many as 3,800 Tyrannosaurus, whereas an area the size of Washington, D.C. might have held only 2 adults. These estimates focus solely on adults, leaving out the younger Tyrannosaurus, which likely occupied different ecological roles, meaning the total population including juveniles was probably much higher. Researchers also note that population densities vary widely among modern predators of similar size. Jaguars and hyenas, for example, weigh about the same but have very different population densities. Additionally, the study highlights the incredible rarity of fossilization. Only about 1 in 80 million Tyrannosaurus may have been preserved as a fossil, though in areas with dense populations the odds might have been as high as 1 in every 16,000 individuals. Not everyone agrees with these population estimates. In 2022, Merry questioned their reliability, pointing out that uncertainties in factors such as metabolism, body size, survival rates by age and sex, habitat requirements, and range size were not fully accounted for. Marshall and his team responded by acknowledging that their reported uncertainties might indeed have been too narrow, but emphasized that their model is flexible enough to include physiological uncertainties. They explained that their calculations rely on long-term population averages rather than short-term changes in density or range, and that they had already factored in uncertainties in the onset of sexual maturity and in growth curves, including maximum body mass. Allosaurus was a fearsome meat-eating dinosaur that lived a very long time ago, roughly 155 to 145 million years ago, during the final chapter of the Jurassic period. Scientists call this time the Kimmeridgian to late Tithonian ages. It belonged to a group of dinosaurs known as theropods, which were mostly bipedal predators. The first fossils that could be clearly identified as Allosaurus were described in the year 1877 by the American paleontologist Othniel Charles Marsh. He gave it the name Allosaurus, which means different lizard. Marsh chose this name because he thought the dinosaurs' vertebrae were unusual. They were surprisingly lightweight compared to those of other dinosaurs. Allosaurus is a genus that has puzzled scientists for a long time because of its complex classification, but at least 3 species are considered valid today, with Allosaurus fragilis being the most famous. Most of the fossil remains of this giant predator have been found in the Morrison Formation of North America, though fragments have also been discovered in Portugal, in places such as Alcobarça, Bombarral, and Lourinhã. For more than 50 years of the 20th century, the species was known under a different name, Antrodemus, until a detailed study of the rich fossil beds at the Cleveland Lloyd dinosaur quarry restored the name Allosaurus. Because it was one of the first well-known large carnivorous dinosaurs, Allosaurus has fascinated both scientists and the general public for over a century. In life, Allosaurus was a formidable predator. It walked on 2 strong legs and had a large, light but sturdy skull, filled with dozens of sharp, serrated teeth capable of slicing through flesh. The average Allosaurus fragilis measured about 8.5 meters long, which is nearly 28 feet, while the largest individuals may have reached almost 10 meters, or 32 feet in length. Its 3-fingered hands were relatively small compared to its muscular legs, but its body was well balanced by a long, powerful tail. Classified in the family Allosauridae, it was the most common large predator of the Morrison Formation and ruled the prehistoric food chain. Its diet likely included enormous plant-eating dinosaurs such as ornithopods, stegosaurs, and even young or weakened sauropods. Scientists still debate whether Allosaurus hunted in groups, showing some form of social behavior, or if it was mostly a solitary hunter that occasionally gathered with others. Evidence exists to support both possibilities. The story of Allosaurus's discovery is tied to the famous Bone Wars, a fierce rivalry between Marsh and another American paleontologist, Edward Drinker Cope, which led to a rush of fossil discoveries across the western United States. The very first bone later linked to Allosaurus was collected second-hand in the year 1869 by Ferdinand F. Hayden near Granby, Colorado, probably from rocks of the Morrison Formation. Locals had thought the bones were petrified horse hooves. Hayden sent the fossil to Joseph Leidy, who identified it as part of a tail vertebra and tentatively assigned it to a European dinosaur called Poecilopleuron, naming it Poecilopleuron valens. He later decided it deserved its own genus, which he named Antrodemus. The genus Allosaurus itself is based on a small group of fragmentary bones catalogued as YPM 1930. These included parts of 3 vertebrae, a rib fragment, a tooth, a toe bone, and the shaft of the right humerus, or upper arm bone. Marsh formally named these remains Allosaurus fragilis in the year 1877. The word fragilis is Latin for fragile, again highlighting the unusually light structure of the vertebrae. The bones were found by 2 of Marsh's fossil collectors, Benjamin Mudge and Samuel Williston, at Felch Quarry in Garden Park, Colorado, in the autumn of 1877. Marsh was not entirely satisfied with the quality of these fossils and briefly closed the quarry that same year. Despite this, he named 2 new dinosaurs based on the finds, Allosaurus and Diplodocus. In the year 1883, he hired Marshall P. Felch, the quarry's original discoverer, to continue excavations. Felch's work turned the quarry into one of the most productive sites in the Morrison Formation, producing holotype specimens of Ceratosaurus, Stegosaurus, and a nearly complete Allosaurus skeleton, catalogued as USNM 4734, which would later become the neotype specimen of Allosaurus fragilis, the main specimen on which the species is now officially based. In the year 1879, a collector working for Cope H.F. Hubble found a nearly complete Allosaurus skeleton near Como Bluff, Wyoming, but it was never unpacked while Cope was alive. It was only in the year 1903, years after Cope had passed away, that the skeleton, now catalogued as AMNH 5753, was unpacked and prepared for display. By the year 1908, it was publicly exhibited as a dramatic mount, posed over a partial Apatosaurus skeleton as though scavenging, famously depicted in a painting by Charles R. Knight. This was the first freestanding mount of a large theropod dinosaur, and while it became iconic, it has never been formally described in scientific literature. The naming of Allosaurus was further complicated by the many rival names proposed by Marsh and Cope, often with very brief descriptions. Even at the time, paleontologists like Samuel Williston warned that too many names were being created unnecessarily. In the year 1901, he noted that Marsh had not been able to clearly distinguish Allosaurus from a genus called Creosaurus. The first major attempt to sort out the naming confusion was carried out by Charles W. Gilmore in the year 1920. He argued that the tail vertebra named Antrodemus by Leidy could not be reliably distinguished from Allosaurus, and suggested that Antrodemus should be the preferred name due to its earlier origin. For over 50 years, the genus was known as Antrodemus until James H. Madsen studied the Cleveland Lloyd fossils and argued that Allosaurus should be used because Antrodemus was based on poorly preserved material with little geological information. In the year 1909, Earl Douglas from the Carnegie Museum discovered a rich fossil site in what would later become Dinosaur National Monument in Utah. Over the years, his team excavated more than 320,000 kilograms of fossils, including many Allosaurus specimens. One of the most notable skeletons, CM 11844, collected between the years 1913 and 1915, included much of the body and a partial skull, and has been on display at the Carnegie Museum since the year 1938. In the summer of the year 1924, the University of Utah unearthed specimen Dino 2560, which at the time was the best preserved Allosaurus ever found. Its particularly large skull is now on exhibit at Dinosaur National Monument. The story of Allosaurus, one of the most famous and well-studied predatory dinosaurs, is closely tied to some of the most remarkable fossil sites in North America and Europe. Work at what would later be called Utah's Cleveland Lloyd Dinosaur Quarry began sporadically as early as the year 1927, and the site was first formally described by William L. Stokes in 1945. However, large-scale excavations did not truly take off until 1960. From 1960 to 1965, a massive collaborative project led by paleontologist James A. Madsen brought together nearly 40 institutions. During these 5 years, thousands of dinosaur bones were carefully removed from the quarry. What makes Cleveland Lloyd so extraordinary is the overwhelming dominance of Allosaurus remains. The site has fossils representing at least 73 individual dinosaurs, of which 46 or more are Allosaurus fragilis. The sheer abundance and quality of these fossils have allowed scientists to study this dinosaur in remarkable detail, making Allosaurus one of the best-known theropods of all time. Fossils from the quarry include individuals of almost every age and size, ranging from tiny juveniles under 1 meter long to massive adults reaching up to 12 meters in length. Because Allosaurus fossils are so common in Utah, both at this quarry and elsewhere, it was officially named the State Fossil of Utah in 1988. In the early 1990s, a Swiss team led by Kirby Seiber began commercial excavations at the Howe Ranch Quarry in Wyoming. This site had originally been explored in 1934 by the famous fossil hunter Barnum Brown and his crew, who collected more than 30 tons of bones, mostly belonging to giant sauropods. The Swiss team could not locate many additional fossils at the original quarry, so they began exploring the surrounding area. Their search led to the discovery of a spectacular specimen in 1991, later nicknamed Big Al and catalogued as Moore 693. This fossil was an exceptionally complete Allosaurus, about 95% preserved, with bones still partly in their natural positions. Because the new site was on public land, excavation was taken over by a joint team from the Museum of the Rockies and the University of Wyoming Geological Museum. Big Al, now displayed at the Museum of the Rockies, belonged to an individual roughly 8 meters long, smaller than average for an Allosaurus, as it was a subadult estimated to be only 87% grown. The Swiss team later found a second Allosaurus, nicknamed Big Al II, SMA 0005, on private land at Howe Ranch, which is now exhibited at the Athol Dinosaur Museum in Switzerland. The classification of Allosaurus species has been a subject of debate for decades. In 1991, Brooks Britt suggested that there were at least two species, a robust form with a short, tall skull and pointed lacrimal horns, and a more gracile form with a long, low skull and rounded lacrimal horns. The robust species appeared in geologically younger sites like Dry Mesa Quarry and Garden Park, while the gracile form was found in older sites such as Cleveland Lloyd and Dinosaur National Monument. Interestingly, as early as 1988, Gregorius Paul had made a similar distinction, naming the gracile species Allosaurus fragilis and the robust species Allosaurus atrox, based on a species originally described by Othniel Charles Marsh as Creosaurus atrox. Later statistical analyses by David K. Smith, conducted between 1996 and 1999, suggested that these differences might simply reflect individual variation within the species rather than separate species. Allosaurus is not limited to North America. Fossils from at least 3 sites in Portugal, within the Lourinhã, Bombajal, and Alcobaça formations, show that this dinosaur also roamed Europe. The first Portuguese specimen, MN Schnull and 001, a partial skeleton including a hind limb and pelvis still in articulation, was found in 1988 near the village of Andres during the construction of a warehouse. Reported in 1999 and assigned to Allosaurus fragilis, it became the first Allosaurus ever discovered outside North America, making Allosaurus fragilis the first dinosaur species known from both Europe and North America, hinting at faunal exchanges between the continents. Further excavations at the Andres site between 2005 and 2010 revealed at least 2 more Allosaurus individuals, including numerous skull bones. In 2005, a single maxilla from the Guimarote coal mine, a site famous for fossils of Mesozoic mammals, was also assigned to Allosaurus, though not to any specific species. In 2006, Octavio Mateus and colleagues described a partial skull and 3 neck vertebrae, ML 415, from Vale Fredas Beach in Lourinhã. Because this specimen showed differences from North American Allosaurus fossils, they proposed a new species, Allosaurus europaeus, and also assigned the Andres specimen to it solely because it was found in Portugal. This suggestion implied that faunal exchange between Europe and North America might have been interrupted, possibly by the opening of the Atlantic Ocean. However, the validity of Allosaurus europaeus has been debated ever since, with some studies suggesting it is simply the same species as Allosaurus fragilis, others labelling it a nomen dubium, and yet others calling for re-evaluation.